and anti-inflammatory effects. First of all, I would like to give some general information. Creatine, lead, that is PB, an environmental pollution and toxic agent, is among the heavy metals. Contaminated food, water, and air pollution are major source of PB toxicity. PB excretion is very difficult and can be stored in soft tissues, bones, and other important organs for a long time. Also, it has toxic effects on many tissues. Creatine, that is CHR, natural found in patient's fruit, honey, and propolis, is a flavonoid, a member of the flavone class with 15 carbon skeletons. Creatine's antioxidant, anti-inflammatory, anti-cancer, anti-diabetic, anti-allergic, and anti-apoptotic properties have been the focus of many studies. There is no definite information in the literature on whether CHR has a protective effect against gastric damage caused by lead. That's why the present study was conducted to investigate the protective effects of CHR on gastric damage caused by lead acetate. For this purpose, animals were divided into 5 groups as control: CHR alone, lead acetate alone, lead acetate plus 25 milligram kilogram CHR, and lead acetate plus 50 milligram kilogram CHR. The study took 7 days. At the end of the study, stomach tissues were taken from the animals. The mRNA transcript levels of the genes presented, table 1, were analyzed by real-time PCR method in stomach tissues. Table 1, genes. Nuclear factor erythroid 2 related factor 2, that is NRF2, is a transcription factor triggered by oxidative stress. Hemoxygenase, that is HO1, is regulated by NRF2. On the other hand, excessive oxidative stress suppresses NRF2. According to the obtained data, it was observed that lead acetate suppressed the NRF2 and H1 expressions. However, CHR treatment upregulated the expression of these genes in a dose-dependent manner. Oxidative stress is an important trigger of inflammation. Inflammation, on the other hand, causes damage to the tissues and causes their functions to be lost. MAPK14 activates NF-kappa B. NF-kappa B, on the other hand, stimulates the release of pro-inflammatory cytokines such as TNF-alpha and IL-1 beta. In the study, it was determined that lead acetate increased the mRNA transcript levels of MAPK14 and NF-kappa B. Probably due to this, TNF-alpha, IL-1 beta, inos, and COX-2. COX-2 expression were upregulated. However, CHR treatment dose-dependently suppressed the expression of these genes. Taken together, it was determined that lead acetate could cause damage by causing oxidative stress and inflammation in the gastric tissues of rats. On the other hand, CHR treatment could protect the gastric tissue from the destructive effect of PBAC, lead acetate. Thank you for your attention and patience.
Thank you, Dr. Gür, for your presentation. Is there any question or any addition? No? Okay, then we can start the second oral presentation. Evaluation of protective effect of Neurangine in Oxaloplatin induced cardiotoxicity via apoptosis and endoplasmic reticulum stress pathway. Dr. Mustafa İleritürk. Yes, Dr. Mustafa.
Hi, dear participants. Açamadım. I'm trying to connect my video connection. Sesim geliyor mu hocam?
Evet, geliyor hocam.
Start my video. Nasıl? Görünmüyor şu anda. Nasıl paylaş? I cannot share my screen. How can I do that?
You can use the green button at the bottom of your screen. Can you see? It is
Yes,
the
I
share
can.
screen.
Okay. Allah.
We can see now your presentation.
You can see. Okay. Hi, dear participants. I'm Mustafa İleritürk from Atatürk University, Department of Biochemistry. My study is called Evaluation of Protective Effect of Norengine in Oxaloplatin-induced Cardiotoxicity via Apoptosis and Endoplasmic Reticulum stress pathway. Chemotherapy is a common treatment primarily used for various types of cancer. However, it can also cause side effects such as cardiotoxicity in cancer patients. Oxaloplatin, a third-generation platinum-based chemotherapeutic agent, is widely used in certain cancer types such as colorectal, prostate, and testicular cancer. Norengine is a promising flavonoid whose cardioprotective effect has been proved in other studies. So these studies aim to examine potential protection of Norengine, which is a well-known antioxidant in oxaloplatin-induced cardiotoxicity. The study groups was made as listed in this slide. For this purpose, 35 Sprague-Dawley male rats were randomly divided into 5 groups. The control group rats were given 5% dextrose solution on the first, second, fifth, and sixth day. Norengine group, 100 milligram per kilogram Norengine was given orally to the rats on the first, second, fifth, and sixth days. The oxaloplatin group, 4 milligram per kilogram oxaloplatin was injected to rats IP on days first, second, fifth, and sixth. The oxaloplatin plus Norengine 50 group on the first, second, fifth, and sixth days, rats were injected with 4 milligram per kilogram oxaloplatin IP. 30 minutes after that, 50 milligram per kilogram Norengine was given orally. Oxaloplatin plus Norengine 100 group on the 1st, 2nd, 5th, and 6th days. Rats were injected with 4 mg per kilogram oxaloplatin IP. 30 minutes after that, 100 mg per kilogram Norengine was given orally. At the end of the experiment, the rats were decapitated under mild sevoflurane anesthesia and heart tissues were removed. Apoptosis and endoplasmic stress markers were analyzed by real-time PCR method in the obtained tissues, which was powdered with liquid nitrogen. This is my real-time PCR method according to Livak and Schmidtgen. This table shows the primary sequences of the evaluated parameters. As you can see, we evaluated 7 parameters. The first of them is CASPAS-3. The figure shows that CASPAS-3, which is well-known marker of apoptosis, was upregulated through oxaloplatin administration. On the other hand, Norengine co-administration decreased that apoptotic effect and reduced CASPAS-3 mRNA expression levels. Similarly, BAX, which is the other important marker of apoptosis, increased in the oxaloplatin group, and Norengine co-administration decreased that expressions. Anti-apoptotic protein BCL2 decreased with the oxaloplatin administration, unlike CASPAS-3 and BAX. And moreover, BCL2 gene mRNA expression levels has approached to the control group with the dose depend in Norengine administration. As you know, endoplasmic reticulum stress pathway consists of 4 different parameters: ATF6, PERK, IRE1 alpha, and GRP78. They play key role in the endoplasmic reticulum stress pathway. These parameters increased in the oxaloplatin administrated group and decreased oxaloplatin plus Norengine dose groups. As you can see, this is ATF6 figure. This is PRK figure. This is IRE1 alpha figure, and this GRP78 figure. All of them are increased with the oxaloplatin administration and decreased with Norengine co-administration. The result of the present study demonstrates that Norengine exhibits protective effects against oxaloplatin-induced cardiotoxicity. Norengine's anti-apoptotic and endoplasmic reticulum stress reducing properties can be considered as the primary factors responsible for its protective effect against cardiotoxicity. On the other hand, further studies are needed to support these findings before a clinical application can be recommended. Thank you for listening. Any questions? That's it.
Thank you very much, Dr. Mustafa. Thank you very much. Is there any question or any addition for this presentation? No? Okay. Thank you, Dr. Mustafa. Now we can start the third oral presentation, obtaining and research of ABS polymer composite materials from Dr. Kamala Badalova. Yes, Dr. Badalova.
Greetings. Merhaba, hepinize merhaba. Ben Türkçe başlayacağım, sonra icazet verirseniz İngilizceye geçeceğim.
Tabii.
Hepinize merhaba, hoş gördük ve davetinize göre çok teşekkür ederim sizlere. Yəqin ki benim slaytlarımı görür müsünüz?
Evet, başladı.
Görülür mü?
Evet, evet. Buyurun.
Okay, now I will start to my presentation. My name is Kamala Badalova. I am assistant professor of the Azerbaijan Medical University. And my topic of training and the research of antibacterial acrylonitrile butadiene stearin polymer composite materials. Okay, but first of all, I will introduce what is composite material. A materials system composed of two or more physically distinct phases whose combination produce aggregate properties that are different from those of its constituent, or two or more chemically distinct materials which, when combined, have improved properties over the individual materials. Composite could be natural or the synthetic. Okay, but which important properties of composite materials we have? First of all, composites can be very strong and stiff, yet very light in weight. So ratios also strange to weight and the stiffness to weight are several times greater than steel or the aluminum. So reticulum properties are generally better than for common engineering metals. And hardness is often greater too. But composites can be designed that don't corrode like steel, possible to have combinations of properties not attainable with metals, ceramics, or the polymers. But one possible classification of the composite materials we have two.
Hocam pardon, çok pardon. Slaytlar ilerlemiyor ama görüntü devam etmiyor slaytlarda.
Sorry, but I will again. Now, can you see?
Evet, tamam şimdi oldu.
Okay, we have two types of composite materials. They are traditional composites and the synthetic composites. But what about the traditional composites? Composite materials that occur in nature or have been produced by the civilizations for many years. Examples wood, concrete, asphalt. But what about the synthetic composites? They are modern materials, systems normally associated with the manufacturing industries in which the components are first produced separately and then combined in a controlled way to achieve the desired structure, properties, and part geometry. Okay, about the development of the composite materials, where we can use about the development of the materials for fuel cell applications, development of the antibacterial materials, development of the biomaterials. They are, for example, dental implants or other materials which we can use in the medicine, and development of the tissue scaffolds. But it is known that the antibacterial properties of the polymer materials used in medicine, agriculture, household products, and many other fields are considered to be one of the most important conditions. One of the technologies for the production of this type of polymer materials is the inclusion of natural and the synthetic antimicrobial additives in polymer composite materials. During processing, many organic and inorganic compounds, oligomers, and the polymers are used in antibacterial additives, and there are many reagents for these additives, one of which is that they must be harmless to the humans. Okay, it is important that the polymeric materials used in various fields are harmless to the human body, as well as resistant to the effects to widespread bacteria and the microfungi. The development of the technology for the production of such antimicrobial polymeric materials, this is APM, is carried out in several directions, one of which is the inclusion of the natural and the synthetic antimicrobial additives in the processing of the polymeric materials. But from this point of view, salicylic acid and its derivatives, as well as oligomers containing a salicylic group, are of the great interest. But in our previous studies, we had before experimental work, and we studied antibacterial composite materials were obtained using allyl and vinyl esters of the acetyl salicylic acid from the biopolymers as a biostol additive. And it was found that composite materials containing the obtained salicylic group have high antibacterial property. And about the physical, mechanical, and the antibacterial properties of these antibacterial composite materials based on the ABC, acrylonitrile butadiene stellar copolymer, were studied using salicylic acid as an antibacterial additive. Composite material, this ABC composite material is a substance with the viscosity of 180 degrees of Celsius and containing 30% ABC copolymer and 70% calcite mineral. This is calcium carbonate. And primary ABC is a substance soluble in ketones, ethers, acetone, and 1,2-dichloroethane and ethyl acetate. And the composition of the obtained composite material is as follows. To prepare composites, acrylonitrile butadiene styrene and the salicylic acid with the antibacterial properties are mixed and extruded at 170 degrees Celsius. And then it is pressed under the pressure of 15 megapascal into a standard plate. Studies have shown that addition of 0.18 and 0.8% solution of the salicylic acid to the composite material has practically no effect on its physical and mechanical properties, but increases the thermodynamic properties with an increase in the amount of additive. But our tests had been carried out, for instance, against the bacterial and the microfungal effect of the new composite material. Microorganisms such as Asparagus niger, then Autologous, Penicillium caclopium, and the Penicillium chrysogenum were used as a test cultures in the study of the fungicidal properties of the antibacterial composite materials based on ABC. But the investigated composite materials were placed in the nutrient medium, bleached malt juice for 30 days, and the mushroom was grown on it. And at the end of the period, the plastic material was microscopically inspected and compared with the original condition. It turned out that none of the tested plastic materials changed due to visual or microscopic images, which can be considered as an indicator of their resistance to fungi. Okay, in end of my speech, I want to show you my country. This is my capital of Azerbaijan, Baku, where you can see the monument, this old monument by name is Gizgalası. This is our mosque, Haydar Aliyev Mosque. This is our inner city, or the old Baku, we can, and also the picture from the inner city with the old our, for example, you can see, for example, by name is samovar, from which we can drink tea. And this is modern Baku. This is building of my university, Azerbaijan Medical University. We have a lot of buildings of the university. This is me with my students, from the different foreign countries. Also, among these different countries, we have students from Turkey, from Iran, from Iraq, from Algeria, from India, from a lot of different countries. And they are students from Africa, from Iraq, from Algeria, Sudan, and other. And also in my workplace, I was Erasmus Mundus media student in Spain, in Santiago de Compostela University, and this is my workplace with ICBMS. Okay, teşekkür ederim. Thanks a lot for kind attention.
Thank you very much for your detailed presentation. Is there any question or addition to Dr. Kamala? Okay, thank you, Dr. Kamala. Thank you very much. Have a good day. And now we can start the third title, Neuroprotective Effect of Curcumin, Elevating Cadmium-Induced Neurotoxicity and Neurogenic through CREP DNF signaling in Swiss albino mice. And Dr. Mariam Sarwat. Yes, we can start. Welcome.
Yes, good morning, everyone. I'm Dr. Mariam. I'm Associate Professor at Amity University, India. And I am grateful to the organizers for giving me this opportunity to present my work in the Second International Congress on Biological and Health Sciences. And I'm very delighted to present my work amongst the august audience today. And Turkey is very close to my heart, so I'm very happy to present. So my work today is about the neuroprotective effect of curcumin. Curcumin belongs to turmeric, which is used in the food. And we have seen that it elevates the cadmium-induced neurotoxicity. And it also promotes neurogenesis. And we have seen that the CREB-BDNF signaling pathway is involved in it when we have studied this in the Swiss albino mice. Okay. So this is a part of the PhD thesis of one of my research scholar, Mr. Dondub Namgyal, who has recently awarded his thesis in December 2021, and he has now joined a postdoctoral fellowship in South Korea. So I'm very proud of him. And these are some of the research papers which we have published, my student Namgyal, and this is very good impact factor in 4.2, 6.6. This is also, these have been published in 2020 and 2021 only. So the objective of the present study is to estimate the neuroprotective effect of curcumin and to analyze the effect of curcumin on neurogenesis. So first, I would like to give you some introduction, like plant-based medical treatment has long history along with the human evolution. According to the WHO, more than 80% of the people from Asian and African countries use plant-based medicine for the primary treatments of diseases. More than 25% of modern drugs have also been derived from the plants, and there are more than 7,000 plant-derived compounds in the modern pharmacopoeia. So these all medicinal properties of these plants are aligned with the secondary metabolites which are present in these plants. They can be phenolic compounds, alkaloids, terpenoids, and sulfur-containing compounds. Now coming to the turmeric use has nearly 4,000 years in the Asian countries. The phytochemical constituents of the turmeric are shown here. And these are the carbohydrates, 70%, water 6 to 13%, fat 5 to 10%, protein 6 to 8%, dietary minerals, essential oils, dietary fibers, and a very important component that is curcuminoid, that is 1 to 6%. So curcumin is a major constituent of this curcuminoid, and it considered as a storeroom of dietary fiber, magnesium, potassium, iron, and vitamins. The medicinal properties of curcumin are anti-inflammatory, antioxidant. It also boosts the cell's defense mechanism. The slides are stagnant.
Doesn't work, or?
Yeah, I'm trying. I think the slideshow mode is creating some issue. So I can present like that. Are you able to see it?
Yes, okay, okay.
Okay, so now coming to the chemical that is CD-induced stress and neurotoxicity. How does cadmium induce it? Like cadmium is a highly toxic heavy metal, one of the most important occupational and environmental pollutants. And a very important thing is that it gets accumulated in the living system, in the humans as well as animals. Cadmium reaches into the environment by the anthropogenic mobilization and it absorbed from tobacco consumption as well or injection of any contaminated substances. A very important aspect of this is that it has extremely long biological half-life. It remains approximately 20 to 30 years in humans, and it has a very low rate of excretion from the body. So it can cause a lot of damage to the soft tissues like liver, kidneys, and etc. It has a diversity of toxic effects such as nephrotoxicity, hepatotoxicity, endocrine, and reproductive toxicities also. Cadmium-dependent neurotoxicity has been also related to neurodegenerative diseases. It can cause neurodegenerative diseases also like Alzheimer, Parkinson, lateral sclerosis, and multiple sclerosis as well. So what is the effect? How cadmium affects? It affects the cell proliferation, the differentiation, the apoptosis, and as well as other cellular activities. Cadmium-dependent interference in the DNA repair mechanism. It also affects the repair mechanism of the DNA and it generates the reactive oxygen species. And this is the most important causes of its cellular toxicity. As we can see in this figure also, the cadmium alters the intracellular concentration of the calcium. So it perturbs the calcium homeostasis. And that is why it affects the Krebs and other signaling pathways, which we have studied in our research. So the rationale of our research was exposure to the various stressors like cadmium has become unavoidable in our modern urban societies. And this stress relates to the neurological disorders, which are rapidly increasing nowadays. Many synthetic drugs which have been used for the cures are associated with various side effects. The herbal medicines, which focus on the overall health rather than the individual diseases, are very much in demand. So in this study, we have studied the pleiotropic health effect of the curcumin. The previous research of various scientists have also shown that curcumin has therapeutic potential for neurodegenerative diseases, as well as for heavy metal-induced neurotoxicity. So for our experiment, we have done this experimental design. We have taken 8 groups of animals, and each group we have 7 mice. We have a couple of controls, where we have given normal food and water and only the carboxymethyl cellulose in which we have dissolved our drug. Then we have taken a cadmium only group, which is the negative control, and then cadmium plus pioglitazone 30, that is the positive control group. And in addition to this, we have taken cadmium along with various doses of curcumin to see what is the effect of curcumin on the cadmium-induced neurotoxicity. So in the other diagram, we have shown that how we have given these doses. This research methodology consisted of about 8 weeks. After the 8 weeks, we have done our studies, various studies, which we will be discussing in the subsequent slides. In the first 4 weeks, we have given only the cadmium in the cadmium groups, and rest of them we have given the cadmium plus curcumin. After 8 weeks, we have done the studies like the behavioral studies, the protein analysis, the biochemical analysis, and etc. And after that, we have sacrificed the mice and studied other things, their histopathology, etc. So now the question is why we have used the pioglitazone here. Because treatment with pioglitazone attenuates the neurodegeneration, which have been already proved, and it improves the cognition in mouse models and patients with mild to moderate Alzheimer's disease. So it has been proved both preclinical and clinical studies that pioglitazone has protective effect on neurodegeneration. It inhibits the neuroinflammation, it facilitates the amyloid beta plaque clearance, and enhances the mitochondrial function and improve the synaptic plasticity. It is how it affects at the cellular level. So these are the parameters which we have studied. We have studied the behavioral test, the biochemical test, the histopathological studies, and the expression studies on the proteins of the hippocampus. So the behavioral studies were divided into open field test, novel object recognition test, and Morris-Water maze test. Now I will tell you why we have chosen these behavioral tests. Like the open field test, or we have also called as OFT, was performed to evaluate the spontaneous locomotor activity of the animals. Like when we are giving the cadmium, what is the effect on the animals, how they behave, how they locomote, how they walk, etc. So this is for OFT, this is the apparatus which being used, which has three concentric circles, which is again divided into quadrants. A video camera was placed on the ceiling light above the arena, and we have recorded the behavior of the animals. Now, what was the behavior which we have recorded? Animals were individually placed in the center of the apparatus, and they're allowed to freely explore all the things with 5 minutes. And we have studied the parameters like total number of crossings from one quadrant to another, crossings in the periphery, crossings in the center, time spent in the periphery, time spent in the center, and the rearing frequency. These are all the parameters of the OFT. Okay, so a crossing was considered only when the animals entered another quadrant with its 4 paws. It means that the animal has entered the another quadrant, then we consider it as a crossing. And after each experiment, we have cleaned it with 10% ethanol water solution so that our experiment will not be affected. So this is the result. After that, we have checked in our all the groups of animals, the control, the cadmium treated, cadmium plus various ranges of curcumin like 20 mg per kg, 40 mg, 80, 160. Then only curcumin and 160 mg, and then the positive control. So what is the inference of this study is that cadmium treated group has lower locomotory activity because cadmium is affecting their neurotoxicity. Okay, so their locomotory activity is affected. Okay, and it as compared to the control and the cadmium plus curcumin 160 group. So curcumin has saved those animals. You can see it from here, and it is a dose dependent effect. When the dose of the curcumin is increased, their activity is restored. And this is comparable to the positive control. In the Morris-Water maze test, another behavior test, we have studied the cognitive functions. The mice were placed in the large circular pool here. Okay, these are the dimension which has been prescribed. Okay, and this is filled with warm water, that is the normal water, with a depth of 30 centimeter. And there is a target platform which is hidden 2 centimeter below the surface of the water in the first quadrant here. You can see. For 5 days, this experiment is going on. Subsequent to drug treatment, we measure the acquisition of memory retention and the cognitive functions, how the cadmium is affected, how is affecting the memory functions, and how the curcumin is affecting those. So in the 4 trials per day, each mouse was randomly released in the pool facing a wall at one of the 4 points, east, west, south, and north direction. We have given the mice into the pool, and they were allowed up to 90 seconds to find the invisible platform. Upon locating the platform, the animals was permitted to remain on the platform for 10 seconds before the next trial, so that they will be knowing where is the platform exactly. The average latency to find the platform and the percentage of time spent in the correct quadrant was recorded. On the last day, after 4th day, on the last day, a probe trial was performed 4 hours after the goal latency test. Using a video recording, we measured the frequency with which the mice were able to find the platform and the average percentage of time spent in the goal platform quadrant. So it means we were training the animal to go and find the perfect quadrant for the perfect platform. So whether they have retained the memory or not, we have checked in various groups of animals. So we have checked the spatial learning and the retention memory through this. So these are again the same groups, 8 groups, which we have discussed. CD treated mice takes longer time to find the hidden platform. This is much obvious. They have taken a longer time to find the hidden platform as compared to the control and the treatment group, because their neuropotential has been affected. CD treated mice spent lesser time in the platform quadrant compared to the control. So they are affecting the lesser time in the platform quadrant because they don't know that this is the right quadrant where we have to be there. So what we have inferred from this is that cadmium has distorted the learning capacity and retention memory, whereas curcumin had restored the distortion. Curcumin recovered the spatial and retention memory dose dependently. The dose is increasing, so ever the retention of the memory has been increasing. Another behavioral test, that is novel object recognition test, is being done. It assesses the learning and memory in mice by familiarizing them with an object and then presenting them with both a novel as well as the familiar object. So by measuring the amount of time spent exploring the novel object compared with the familiar object is recorded. So for them, they are square-shaped open field boxes with objects located at opposite sides of the starting point. Here you can see this. So they are the familiar object which has been given in the training phase, and in the testing phase, there is one familiar object and one novel object is being given to the animals. So mice were allowed to explore these two identical objects in the open field for 10 minutes. That is called as the learning period or the training phase. After 24 hours interval, mice were allowed to explore one familiar object and one novel object as part of the learning period protocol here. The time spent exploring the familiar and the novel objects and the movement track of mice were recorded by a tracking system. So this is the result which we have obtained. So we have calculated T2 upon T1, that is time spent with the novel object and time spent with the familiar object. So we have seen that cadmium treated mice here, you can see has lower T2 upon T1 ratio as compared to the control and curcumin treated group, as you can see here. So what is the inference? Cadmium treated group showed weaker recognition memory. Their recognition memory has become weak due to the effect of cadmium.
Dr. Mariam, can
yes.
we be more fast, please? Because
sure,
I will. Much, attention, because thank you.
Sure. Okay, so curcumin recovered the impaired recognition memory dose dependently. And these are the results of the biochemical test. We have given the SOD, we have checked the SOD, MD, and GSH levels here. So the results show that cadmium treated group has lower superoxide dismutase and catalase activity compared to the control group. Curcumin treated group shows increased enzyme activity, which is dose dependently. So what is the inference from the biochemical test is that CD induced neurotoxicity through the oxidative stress. Curcumin had dose-dependent antioxidant effect. Cadmium had promoted the oxidative stress through the production of pro-oxidant and suppressing the antioxidant enzyme level. Curcumin had protected the CD-induced oxidative stress. This has been proved through these experiments of the biochemical tests. Now, when we sacrifice the animals and we have studied their histopathology, we have checked that how much their neurons are dead cells and how many are the viable cells. So what is the inference from the histopathology is that cadmium exposure induced the degeneration of hippocampal neurons, and curcumin promotes the hippocampal neurogenesis. So what is the molecular mechanism behind that? For that, we have studied various neurodegenerative markers like BDNF, brain-derived neurotrophic factor, synapsin 2. Okay, so when we studied the BDNF and synapsin 2, what is the result is that cadmium treated group showed significant low level of BDNF and synapsin 2 proteins compared to the control group, whereas curcumin had increased the BDNF and synapsin 2 protein dose dependently, as you can see. So what is the inference is that cadmium induced neurotoxicity through down regulation of BDNF and synapsin 2 protein synthesis, and curcumin has protected the neuronal damage through these mechanisms. Subsequently, we have studied the DCX, double cortin protein, and the cyclic AMP protein, CREP protein also as the neurodegenerative markers, and we have seen the low levels of DCX and CREP protein in the cadmium groups. And when we have given them curcumin, they have upregulated the DCX and total CREB protein. So they are all involved in the signaling process. So what is our conclusion from this study is that cadmium induced the oxidative stress through production of pro-oxidant MDA and inactivation of antioxidant, SOD, GSH, and catalase activities. Stress and depressive-like behavior in cadmium-treated group, which we have seen through the behavioral studies, it might be due to the oxidative stress-induced neuronal damage. Cadmium-induced down-regulation of hippocampal proteins such as BDNF, synapsin, DCX, and total CREB might be linked with the memory loss and weaker memory retention, as well as the anxiety and depressive-like behavior. So DCX protein is used as a marker for neurogenesis. It is clear from our findings that CD hampers the neuronal growth and neurogenesis, while curcumin had effectively improved the behavior of rodents and has also increased the brain hippocampal proteins, the BDNF, synapsin 2, DCX, and total CREB. From this study and previous reports, we could thereby conclude that diet supplemented with curcumin has strong effects on the CNS protection, and curcumin use is indeed a novel approach for treating the neurodegenerative disorders. But thank you for your patient listening, and whatever your question is, I'm ready to answer.
Thank you, Dr. Mariam. Is there any question to Dr. Mariam for this presentation, detailed presentation? Okay, thank you, Dr. Mariam. Now we can start the fifth title, Dr. Meliha Koldemir Gündüz. Yes, Dr. Melia, we can start. Dr. Melia, can you hear me? Maybe there's an internet problem. Ulaş Hocam, dilerseniz herhalde Melih Hanım'ın internet bağlantısında bir sorun oldu.
Sistemde yok mu hocam? Tamam, isterseniz diğeriyle devam edelim hocam. Diğer hocamızla.
Tamam.
Birazdan biz kendisiyle kontağa geçelim.
So we can start the sixth one, the new turn. Analysis of activity of protein engineered lipase variants on different 4-nitrophenyl esters. Dr. Nurcan Vardal Yel. Dr. Nurcan?
Yes, I'm here. Just trying to share my screen. Can you see?
Yes, okay.
Okay, so I can start. So, good morning to all. I am Nurcan Vardar from Altınbaş University, and today I'm going to talk about my last work named Analysis of Activity of Protein Engineered Lipase Variants on Different 4-nitrophenyl Esters. Okay, so first of all, I would like to give little information about the enzymes. As you may all know, that enzymes are biocatalysts that regulate most of the metabolic reactions in the cells. And the microbial, especially the microbial enzymes, are important tools that are frequently used in field of biotechnology. And this microbial enzyme, which which really plays a role in intracellular biological reactions, are used in many different industries, especially in food, textile, detergent, pharmaceutical, or cosmetical industries. And among these enzymes, especially the lipase, protease, and amylase are important members of the microbial hydrolytic enzymes. And if we look at the specification of the engineered enzymes, the aim of this engineering is to improve this enzyme to use in different industries. So the specification of the engineered enzymes, generally lipases hydrolyze oils and fatty acids, and their biotechnological potential is enormous. And due to their resistance to organic solutions and broad substrate specificity or radiospecificity, and the engineered lipase are also resistant to high temperatures and remain stable in some alkaline or acidic environments. So especially this small size of bacterial lipases provides the ease of the use in enzyme studies, and their substrate specificity makes these enzymes valuable in biotechnology fields. And it has many application areas in industry. So, like I said, the engineered lipase has really large areas in industry. For example, biodiesel production, biopolymer synthesis, paper manufacturing, biosensor manufacturing, cosmetic, perfume industry, leather industry, medicine, detergent industry, wastewater treatment, and food and pharmaceutical industry, and so on. There are really large areas in industry. And in this study, the aim of the study be to development of the bioprocess industries with protein engineering methods. And for this purpose, we used the kinetic analysis and characterization of the protein engineered lipase enzymes. And also we used different substrates such as paranitrophenol acetate, butyrate, octanoate, dodecanate, and palmitate substrates to determine this enzyme activity. And these substrates actually, the length of the substrates is getting bigger and bigger. And we try to understand which substrate give the best results for us. And the first method is the calibration. So we need to calibrate the paranitrophenol substrate. And this environment, in these studies, we used environmentally isolated lipase enzyme was developed by the protein engineering method in our previous study, and it cloned into the E. coli BL21 to use in the study. And we used 8 different concentrations for paranitrophenol substrate solutions, and their absorbance at 405 nanometer was obtained, and the standard curve graphic was drawn according to concentration and absorbance value. And for the protein assay method, we used the Bradford protein method was used to determine the amount of the protein for both the high activity tubes collected from the column and the homogenate, and ready to use Bradford reagent containing comestible brilliant was used, and the absorbance values against the blank were read at 595 nanometers. And the lipase activity measurement, we used the lipase activity using paranitrophenol substrate determined by the spectrophotometric method by using the microplate reader, macroplate spectrophotometer reader. And the amount of the paranitrophenol released from the paranitrophenol substrate were measured periodically at 405 nanometer at room temperature. And also the acetonitrile was used as a blank solutions. And also 7 different concentration substrates, like I said before, were examined with 10 microliter of enzyme concentrate, and all experiments are repeated for the 3 times. If you look at the kinetic analysis, the initial velocity was calculated from the time paranitrophenol concentration graphic, and the Km and Vmax values were determined with the help of the Landweber-Burke plot and also Michaelis-Menten graphics, and the wild type and also different variants were compared to obtain the relative results. And in this results, in this study, the absorbance of the paranitrophenol products formed as a result of the degradation of the paranitrophenol esters by the effect of lipase enzyme. And this released paranitrophenol was measured spectrophotometrically. The activity of the same lipase enzyme on substrate of different carbon lengths was investigated. And in addition, the effects of lipase variants with different activating properties on similar substrates were examined and compared. And five different paranitrophenol esters were used in this study. As you can see in this figure, these are the substrates: paranitrophenol acetate, which is the shortest substrate, and butyrate, octanate, dodecanate, and palmitate. Palmitate is the longest carbon length. And this figure shows the Vmax, Km, and Vmax /Km values of the lipase enzymes on different substrates. And the Vmax values of the wild-type lipase enzyme increased up to the 8 carbon substrate, and which is the octanate. It's going to increase to the octanate substrate, and decreased with the 12 carbon paranitrophenol dodecanate. The highest value of the Vmax is obtained by the paranitrophenol octanate substrate. And the lowest Vmax value was obtained with the 16 carbon paranitrophenol palmitate, while the highest Vmax value was obtained with the paranitrophenol octanate substrates. That shows this result shows this engineered lipase enzyme with this paranitrophenol octanate substrate give the best results for us. And the activity and the catalytic efficiency of wild type and these 3 different lipase variants, which we called variant 1, 2, and 3, were analyzed spectrophotometrically. And among these variants, and Vmax value of the wild type 2 enzyme was found to be 1.3 times higher than the wild type. Maybe here, wild type 2, we can see this is the Vmax value of the wild type and the variants here. So the variant 2 is 1.3 times higher than the wild type. And since among this variant, the Vmax value of the since this Km values are similar to wild type, the catalytic efficiency of the Vmax /Km value increased 1.18 times for the wild type, for the variant 2. So this variant gives us the best results. The both Vmax values of wild type 1 and wild type 3 variants were calculated as 0.48 and 0.22 respectively. So like I said, the best results for among this variant were obtained by the variant 2. So, as a conclusion, the importance of the lipase enzyme in industry has been emphasized once again with this study. The activity of the wild type and the variant enzymes were analyzed spectrophotometrically, and the results were compared with the variants. And the catalytic activity of the variant 2, which is the one of the lipase variant developed by the protein engineering from our previous study, was increased compared to the wild type. And we have a publication with this results. This one is published in 2025 at the end of that year, so it's quite new. And what can we do in future work? As we understand the variant 2 gives us the best results, so we are trying to understand what is the genetic changing in these variants. So it is planned to carry out these genetic studies on the basis of the results obtained from these results. And we are trying to determine the region of that enzyme, the variant 2 enzymes, where the mutation is located, and examine the effects of this amino acid change, which amino acid change, what happens in variant 2, and the three-dimensional structure, to understand the three-dimensional structures of the protein. This is our future work. And thank you for listening. I hope I didn't pass my time.
No problem. Okay. Thank you, Dr. Nurcan. To Dr. Nurcan, is there any question, any addition? Okay. Thank you, Dr. Nurcan. Now we can turn back to fifth title. Dr. Melia Koldemir. Bağlanabildiniz mi Melia Hanım?
Evet hocam, hemen paylaşıyorum sunumu.
Okay.
Sorry for losing my internet connection. Hello everyone. I will present my work titled Investigation of Mechanism of Alpha-tocopherol in Reducing Lipid Accumulation in 3T3L1 adipocytes via CTRP3 gene expression. Obesity is a chronic condition in which the quantity of energy consumed by the body exceeds the amount utilized by the body. Obesity is now recognized as a condition that affects all organs and systems of the body, particularly cardiovascular and endocrine system. According to WHO, 1.6 billion people are now overweight, and more than 2.8 million people die each year as a result of being overweight and or obese. Obesity has become into a public health problem, and to fight against it has become a global issue. Considering the high prevalence of and severity of obesity, the number of weight control drugs is much less than other chronic diseases. For patients unable to lose weight with lifestyle change, despite ample evidence of significant weight loss effects and the safety of pharmacotherapy, these drugs are still not used enough. The main reason for this is the side effect of the drugs. These effects develop depending on the dose. In addition to pharmacotherapy, many chemical, herbal, and nutritional supplements are used for the treatment of obesity, the effectiveness of which varies according to the condition, amount, and duration of use of the patient, as well as low-calorie diets and increased conventional physical activity. One such nutritional supplement is vitamin E, whose effect on body weight and mass have been extensively studied. The 3T3L1 cell line is multipatent stem cell line, and these cell lines can transform into cells such as adipose, cartilage, and muscle with the signals coming to them. Modeling obesity in vitro 3T3L1 adipocyte represents a successful system for investigating adipocytic lipid metabolism and adiposity. Vitamin A consists for tocopherol and for tocotrienol isomers, of which alpha-tocopherol and gamma-tocopherol are most commonly found in the diet. Vitamin E, which has an antioxidant effect, plays an important role in energy metabolism associated with the expression of various genes. Alpha-tocopherol contributes to the regulation of genes involved in various cell functions. Alpha-tocopherol has been shown to increase the adiponectin expression through activation of PPR gamma, a master regulator of adiponectin expression. It can control energy balance and weight by increasing adiponectin expression. In addition, vitamin E shows cholesterol lowering effects. C1q tumor necrosis factor associated protein 3 is a C1q tumor necrosis factor related protein that is paralog and adiponectin. CTRP3 is a new adipocytokine that has recently emerged as an important regulatory adipokine of obesity and related metabolic disease. It influences glucose and lipid metabolism, inflammation, proliferation, apoptosis, fibrosis, ischemic damage, vascular calcification metabolism in multiple ways. In this study, the cytotoxic effect of alpha-tocopherol on 3T3L1 adipocytes were evaluated. In addition, the role of alpha-tocopherol in reducing fat accumulation in adipocytes was investigated through gene expression of a new adipocytokine, CTRP3. 3T3L1 fibroblast were pushed from the American ATCC. It was then incubated in a medium consisting DMAM, FBS, and penicillin streptomycin, subcultured as necessary by routine trypsinization method. The proliferation 3T3L1 fibroblast cells were transformed into adipocyte cell. Insulin dexamethasone 3-isobutyl 1-methylxanthine were added to the medium content for differentiation. After observing lipid droplets under microscope, oil red O staining was performed, and adipocyte differentiation was determined morphologically by red staining of oil lipid droplets. Before the administration of alpha-tocopherol, a literature study and daily use were investigated to determine the dose, and the dose range was determined. Alpha-tocopherol solution were prepared mechanically in the medium, and 50, 25, 10, 5, 2.5, and 1 microgram per milliliter were added adipocytes, and the cells were incubated for 8 hours. Only culture medium was added to the control cells. After the application of different doses of alpha-tocopherol, the MTT method was applied as a cytotoxicity test, and the results were analyzed with the GraphPad program. Viability percentages of cells was calculated. After the application of different doses of alpha-tocopherol, total RNA isolation, cDNA synthesis, and qPCR analysis were performed respectively. Expression of CTRP3 and PPR gamma gene was determined by the qPCR using a Stefan Plice instruments. GAPDH as reference gene. Delta CT technique was used to confirm the presence of specific gene products. As a result of alpha-tocopherol application, the cells were tested MTT. The IC50 value of alpha-tocopherol at 48 hours was calculated as 104 microgram per milliliter. This dose was found is lower than the doses applied in the literature. These results show that alpha-tocopherol has a cytotoxic effect on adipocytes. to visualize the lipid droplets after development, the mature adipocyte oil rate was staining was used. Depending on the alpha tocopherol dose, lipid accumulation in the adipocyte was significantly reduced. This showed that alpha tocopherol reduced the lipid contents in adipocytes. This shows that alpha tocopherol can be effective in treatment of obesity. We see the expression of differences CTRP3 and PPR gene, PPR gamma gene as a result of application of IC50, IC25, IC50 and IC78 dose of alpha tocopherol. CTRP3 gene levels is lower in obese individuals compared to control. In this study, CTRP3 gene expression level increased in dose-dependent manner. In adipose cells treated with the alpha tocopherol, increased CTRP3 mRNA levels may indicate inhibition of lipogenesis. PPR gamma gene expression level was decreased. IC50 level 104 microgram per milliliter doses alpha tocopherol application. Alpha tocopherol can inhibit adipogenesis. As a conclusion, in obese individuals, the use of alpha tocopherol with dietary habits or supplements may be beneficial in reducing those related side effects in the use of hybrid drugs in the treatment of obesity by reducing lipid accumulation in adipose sites. And thank you for listening.
Thank you very much for your whole presentation. Are there any questions or addition to Dr. Meliha? Okay, then we can start the next one. Isolation and identification of a novel Pichia occidentalis strain from fermented plant material. Dr. Zehra Erkol.
Yes, I'm here.
Welcome.
Can you see my screen?
Now, okay.
Hi, dear researchers. I'm Zehra Erkol from Abdullah Gül University in Bioengineering Department. I'm undergraduate student, and I'm very happy to be with you, dear professor and dear researchers. It will be a nice experience for me.
Welcome.
During my presentation, I will be talking about. I'm sorry, I forgot the turn of my camera.
Okay.
During my presentation, I will be talking about our study, which had the title of the isolation and characterization of a novel Pichia occidentalis from plant material. As I said, I'm Zehra Erkol. During my presentation, you can interrupt me and ask your question because I will skip some parts due to the time restriction. I want to start with the importance of strain specificity because it explains why we this study, why we conduct our experiments. In a basic term, strain is a genetically specialized subspecies of probiotic microorganisms, and strain selection is vital. I want to give one example. There is the two cars, as you can see. They have the same brand, but they are different features. We cannot expect the same output from these cars, one of them faster than the others, because they are different features. The same situation is valid for the strain, actually. Some strains show the beneficial for human health, but the others cannot. So we need to identify, we need to determine which strain show the probiotic properties, which one is not. I talk about the probiotics. Probiotics, they are living microorganisms. Actually, in our body, we have the good yeast and bacteria as a probiotics. There are health benefits, and there are some of examples, Lactobacillus and Bifidobacterium. are examples of the bacteria. Saccharomyces is the one of the examples of the yeast. Sometimes our body needs additional probiotics, and there are so many different drinks and foods that include this probiotics yeast and.
Zehra Hanım, slaytlar ilerlemiyor ama.
Really, I'm sorry.
Tamam.
Just happened. You can see now?
Tamam, evet.
Thank you for your warning. I'm here. I'll talk about the probiotics and then continue with the fermented plant material. As I said before, there are so many foods and drinks that include the probiotics, yeast and bacteria, and sometimes our body needs these foods and drinks. Shalgam, one of these examples. It is famous in the southern region of Turkey, especially in Adana, and produced in here. It is ancient traditional beverage. It has red colored, cloud and sour soft drink. Raw materials of shalgam are black carrot, bulgur flour, turnip, salt, sourdough, and water. Lactic acid bacteria and yeast have crucial role while forming of this plant material. We use this plant material to isolate our citrine, but before that, I want to just touch this topic, process of shalgam production. There is two different methods. One of them traditional methods, the other is direct methods. In traditional methods, we can see there is two different fermentation steps in first fermentation and second fermentation. On the other hand, in direct methods, there is just one fermentation. We can say that the fermentation is a really important process for producing shalgam. Let me continue with the materials and methods. To isolate of P. occidentalis strain from shalgam, firstly we did serial dilation and we used the YPD medium, agar medium, and our plates were incubated at 30 Celsius degree for 48 hours. And then following step is the strain plate method to pick one colony from our plates. Next step is DNA extraction and PCR amplification. For DNA extraction, we use meshurinaceous nucleosperm microbial DNA kits. The protocol was certainly it's written on it because it is a novel method. And we use different primers, ITS1 and ITS4 primers used for identification of yeast strain, NL1 and NL4 primers for using for the amplifying 26S rRNA region of the microorganism. Finally, we use the M13 primer for PCR fingerprints. You can see the primer and it's sequenced in the table. And this is our gel results. After running PCR samples on the agarose gel, samples that were amplified with the M13 and NL were observed as multiple bonds. The samples were amplified with the ITS primer could be seen on the gel image clearly. The sample that was amplified with the ITS primer had only one clear band on the gel image, and it showed that the specific binding. The next step, we were sent our PCR samples for ITS, Ficus Biotechnology Ankara for Sanger sequencing. We used the ICBH platform and blast analysis were conducted, and our yeast strain were determined by these methods. You can see the table, maybe it's small. Probiotic or not. There is two tests that we conducted by us, but first I want to explain why we did this test. To determine any microorganism is probiotic or not, there are two main criteria for deciding whether a given strain of a microorganism is probiotic or not. One of them is the effect on pH on probiotics in gastrointestinal tract. The second one is the effect of the bile salt. Yeah, this is our acid tolerance test results. Probiotics must be able to survive their passage through the mammalian gastrointestinal tracts, and the probiotic organisms show an increased number of the viable cells in pH 3, and we can see that our results are yeast strain survive at the pH 3. It means that it may have the probiotics properties. and bile salt tolerance test. Bile tolerance, more specifically the tolerance of probiotic microorganism to the bile salt, is an important criteria for determining whether a given strain of microorganism is probiotic or not. Bile function as a biological emulsifier, reducing the surface area of the lipid molecules and so that lipids get easily digested. This is our results. Again, it's confirmed that it may have the probiotics properties. The last step are studies with fermentation. Actually, there's too many details and too many things that we need to talk about. As I said, due to the time restriction, I will skip some parts. These are our reactor parameters: temperature, pH level, PO2 level, and we use the two different media to test our yeast strain can survive in dextrose as a carbon source or dextrose to sucrose, because we know that the dextrose is a little bit expensive carbon source. But when we use the sucrose, it is cheap and it is easily reachable. These are the results. We use the two trial. It shows that our yeast strain can survive in the sucrose to dextrose media. Also, it can live in the dextrose. Actually, we have already know it. It can live in the dextrose. These are the trial, and they are similar with each other. Thank you for your interest. If you are interested in this topic, you can visit our website or our social media account. If you have any question, I will try my best to answer.
Thank you for your presentation. Are there any questions? Dr. Zehra?
I'm not a doctor, actually. I hope I can, but I'm undergrad student.
I know, but I want to say to
Thank
you.
you, thank
You have
you.
very good presentation.
I'm glad to hear that.
Thank you. Have a good day. Now the next one, synthesis, characterization, biological and molecular docking assessment of novel aflatoxin derivatives. Dr. Mohammed Arsat.
Yes, Assalamu alaikum. I am here.
Okay,
How is
welcome.
it? Very good morning.
Good morning.
Yeah. First of all, I want to thank you so much for giving me an opportunity for discussing my topic, research topic in this conference. And this is the second International Congress on Biological and Health Sciences. Okay, so my topic for discussion is synthesis, characterization, biological and molecular docking assessment of novel aflatoxin derivatives. I am working here at Shakra University as an assistant professor in College of Medicine, Dawadmi, Shakra University, Kingdom of Saudi Arabia. Next slide. Slides are not moving. Slides are not moving. Sir.
Ulaş hocam, slaytlar açılmadı ama.
Yeah, now you can see.
Okay.
Yeah. So first of all, I want to just give an overview about the importance of this study, why we have chosen this study. So as per the literature, and we have know that the microbial infections are one of the biggest health hazards worldwide, as it has been reported to be the reason for the deaths of 20,000 people every year in the USA alone. Okay, and this is happening due to the development of resistance for the available antimicrobial agents, and this is one of the reasons for the mortality reported due to these microbial infections. The resistance of pathogens against the antimicrobial agents have been reported, such as methicillin resistant Staphylococcus aureus, penicillin resistant Staphylococcus pneumoniae, and vancomycin resistant enterococci. So it has been reported, and so therefore it becomes the necessity to search some new antimicrobial agents which will be having some different mode of action or with low molecular weights or some different scenario. And on the other hand, we can see that the fluoroquinolones, specifically the fluoroquinolones means ciprofloxacin, ofloxacin, and norfloxacin, So these are all fluoroquinolones, and they are antibacterial agents that are going to attack the DNA gyrase and topoisomerase 4 on bacterial chromosomal DNA, increasing the evidences of fluoroquinolones that they have developed the resistance against various gram-negative bacilli, Pseudomonas aeruginosa, and Escherichia coli, Salmonella, to approach a critical point. Ofloxacin is also one of the antibiotics which have developed so much resistance against the microbial pathogens and having some adverse effects also. Additionally, the derivatives which have some important heterocyclic nucleus like thiodiazole, triazole, ship bases, thiones, oxadiazole, thiazolidinone, and oxadiazolene exhibited versatile pharmacological applications. So keeping in mind all these parameters like the development of antimicrobial resistance and the importance of these heterocyclic nucleus like oxadiazole, thiodiazole, and the resistance developed against ofloxacin and its side effects just prompted us to design some study related to that, and we have designed some compounds taking as a nucleus, ofloxacin as a nucleus we have considered, and we have designed some derivatives. And after designing, we have calculated just preliminarily we have calculated these for computational screening through the available online software, that is mole inspiration, and we have calculated the physicochemical properties and drug likeness. After that, we have synthesized and characterized these compounds and then led to the biological screening, antibacterial, antifungal, and MTT assay to understand the percent viability of the cells. Then after doing this, we have also performed the molecular docking studies. Okay. And the molecular docking studies was performed against the glucosamine 6-phosphatase to understand the level of hydrogen bonding in between the designed and synthesized compounds with the amino acid residue of glucosamine 6-phosphatase. Okay, so here you can see that sequentially I will discuss that what we have done. Now, how we have calculated the drug likeness and physicochemical score. The structures of all the design compounds were designed using ChemDraw Ultra 12.0, and we have just copied the SMILE files from there, and these SMILE files was used on the online available software, that is mole inspiration, and we have calculated the drug likeness, that means the bioactivity score and the physicochemical properties from that. Okay, these are the structures which we have designed, and we have calculated the drug likeness and physicochemical properties. And it has been seen that if the compound, most of the compounds of our series were bioactive because they were having the value from 0 to 0.5. Okay, and 0 to 0.5 is the value of the bioactivity score that is dealing with the active drug molecule. And this was the synthetic protocol which we have used for the synthesis of our designed compounds. Okay, here you can see the conversion of this carboxylic acid means ofloxacin is going to be just a formation of hydrazide and then leading to the ship base formation, and here leading to the formation of oxadiazole, and then here the formation of thiodiazole, formation of triazole, and formation of oxadiazolene, and formation of thiazolidinone. So here now after synthesis of these, because these are bioactive compounds, now we have led to the synthesis, and synthesis was done using the schematic diagram that we have represented in the last slide. Now structure elucidation of the synthesized compounds was carried out using various spectroscopic methods such as FTIR spectroscopy, NMR spectroscopy, both proton NMR and C13 spectra, and mass spectroscopy. The spectroscopic findings strongly confirm the synthesis of compounds, and here you can see just for one compound carbohydrazide we have mentioned FTIR, we are having all the this NH2, the peak for NH2 at 3321 and this 3188, it is going to confirm the synthesis of the design compounds too, and by the FTIR spectroscopy, and for NMR spectroscopy also we can see the peak and the chemical shift at the range of 9.892, that is for specific for NH2. So yes, now antibacterial screening. After synthesis, we have just led to the antibacterial biological screening that we have done for antibacterial, antifungal, and MTTS. The antibacterial screening was performed using disk diffusion method and serial dilution method. The antibacterial findings were collected in terms of zone of inhibition and minimum inhibitory concentration. Okay, so here the 6 mm paper discs were prepared and dipped into the stock solutions of the synthesized compounds, and the zone of inhibition was calculated. The antibacterial screening was carried out against the bacterial pathogen Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus pyogenes, Macrococcus luteus, Klebsiella pneumoniae, Pseudomonas aeruginosa, E. coli, and Proteus mirabilis. So these were the microorganisms that were under the study. So the findings, the antibacterial findings exhibited that the compounds possessed significant antibacterial potential. Okay, and especially if we will see that we have calculated the percent area of inhibition against the reference drug, which was ofloxacin in this study, and the compounds 2, 4, 8 were reported to exhibit better antibacterial potential. It was observed that compound number 2, and compound number 4 and 8 were having better antibacterial activity than the reference ofloxacin used in this. So antifungal screening. Now, the in vitro antifungal screening of the ofloxacin and its analog was performed following the method of serial dilution and agar disc diffusion. The fungal pathogens utilized in the study were including Candida albicans, Candida krusei, Aspergillus flav, and Aspergillus fumigatus, Trichophyton ingredient, and Penicillin burnetii. Okay, the fungal isolates were growing using dextrose, dextrose medium, and the suspension of each fungal isolate prepared by transferring it into 3 cubic centimeter solution. The antifungal findings were also collected in terms of zone of inhibition and minimum inhibitory concentrations, and we have calculated the percentage area of inhibition and plotted against the compound. The antifungal activity findings exhibited that the compounds were having significant antifungal potential. The similar compounds 2, 4, 8 were also representing the better antifungal potential than the reference drugs that were used in the study, amphotericin B. So now, after calculating the antimicrobial activity, we just also performed the MTT assay to understand the percent viability of the cells, to understand the toxic nature of the cells, that these compounds will be toxic, how much these compounds will be toxic towards the cell, and the cells were used in the study were HAP G2 cells. Okay, so to assess the percent viability of the cells, MTT assay was performed against HAP G2 cells. The HAP G2 cells were cultured in DMEM medium, and the percent viability of the cells were recorded by ELISA, enzyme-linked immunosorbent assay. The MTT assay works on the principle that involve the conversion of MTT complex to the formation diet. Okay, and this is observed that the only the compounds which have the live mitochondria, they will be able to convert. They will be able to only the compound formation diet, the help of an enzyme that is situated in the mitochondria of the live cells. That means only the viable cells have the ability to convert MTT complex to the formation, because they have the enzymes, and that is only having the ability to convert this to formation diet. The percent viability of cells were observed concentration dependent. The results of the MTT assay exhibited that the analogs were less toxic against HAP G2 cells. It was observed that these were the compounds which we have synthesized were found less toxic against HAP G2 cells and represented 75 to 80% viability of the cells at the concentration up to 100 micromole. Okay, here you can see also we have also designed this figure again, percent viability of the cells and treated compounds. So as per the concentration, you can see that the concentration, with different concentration we have designed, the concentration is increasing, the percent viability of the cells is also going to be decreasing. So now molecular docking. To understand the state of hydrogen bonding and the binding affinities in between the amino acid residue of the receptor glucosamine 6-phosphatase, we have used the PDB file 2VF5 and the ofloxacin and its derivatives. The molecular docking was performed. The study was accomplished using the softwares like Autodoc tools 1.5, 1.5.6, Autodoc Vina, and the docking images were developed using Taiwan software as per the procedure that we have mentioned in the published article. Okay, the preliminary structures of the compounds were drawn using the software
Dr.
Camera Ultra.
Mohammed, Doctor, can we be more fast, please?
What?
Can we be more fast?
Okay.
Yes, thank you.
Okay, so the preliminary structures of the compounds were drawn using this, and we have used the Autodoc tools, and finally the docked images were received through Autodoc Vina. So now the files were used to develop the docked images by the use of PyMod, and these are the docked images. You can see that how it is going to bind with the. This is the docked images for the compounds which we have synthesized with the amino acid residues of the glucosamine 6-phosphatase. Okay, and the binding energies for these compounds were observed in the range of minus 6.7 to 7.9 kilocalorie per mole. Okay, so here I am just going to conclude this. A series of new ofloxacin derivatives was designed, screened computationally, and led to the synthesis. The synthesis compounds were strongly supported by the spectroscopic methods. The compounds represented the significant antimicrobial, both antibacterial and antifungal potential. The compounds were less toxic, and the molecular docking studies showed that the compounds were having good hydrogen bonding with the residues of glucosamine 6-phosphatase and with low binding energy. Thank you all. Thank you so much for listening. And now, if you have any question, please you can ask. Thank you, sir. Thank you so much.
Thank you very much again, Dr. Mohammed.
Yes.
So, Dr. Mohammed, is there any question or any addition? Okay, thank you, Dr. Mohammed.
Yes.
Now we can start Dr. Nur El Huda Bekkar, therapeutic application of Sisyphus. Dr. Bekkar? Maybe we have an internet problem. Now, okay, the next one. Angelia Ranjan, ameliorative effect of curcumin and soya extract against arsenic toxin Drosophila melanogaster. Angelia Ranjan. Okay, the next one. 11 title. Sana Ketri.
Yes, good morning.
Good morning. Yes. How are you?
Fine, thank you.
Thank you. Okay, you can start. Welcome.
Yes. Just a minute. So, good morning, dear participant. Today, I'm honored to present my work, which is a phytochemical study on the variability of pinus pinester iton, essential oil, and evaluation of their herbicidal and antifungal activity. The objective of this study, first of all, is to indicate the chemical composition of the three aerial part of the species, then to assess the herbicidal effect, and finally to assess the antifungal effect. As we know, all over the world, there is a powerful interest to recommend the plant extract, including the essential oil, to be investigated for their biological activity as a source of biopesticide in order to improve safer alternative. Today, our interest is toward the maritime pinester iton, which is a rapidly growing coniferous species of the Pinus genus Pinaceae family. There is around 115 species of pin, which are divided into three subgenera depending on the character of cone, seeds, and needles. Now, we will start with the chemical composition. Essential oil composition was investigated by GCMS, which is an analytical method that combines the features of gas chromatography and mass spectrometry. The essential oil were a mixture of 5 chemical classes, as shown in the graph here. So we found the hydrocarbon monoterpene class, the oxygenated monoterpene, the hydrocarbon sesquiterpene, the oxygenated sesquiterpene, and the hydrocarbon diterpene. The essential oil were a mixture of 5 chemical classes, included monoterpene with a rate between 32 to 90%. The oxygenated monoterpene was between 0 to 5%. Sesquiterpene was in between 4 to 37. Oxygenated sesquiterpene was between 0 to 7%, and diterpene hydrocarbon was between 0 to 16%. It's clear in the graph that the hydrocarbon monoterpene class was the most abundant class in the 3 essential oil from needles, branches, and cone. Now we will see the chemical composition of each part. For needles, the major component in needles oil were alpha-pinene, which represent 25%. We have also Z-carofilane with 21, and gyramersen D 5%. For branches, alpha-pinene was the major component, and in comparison, which present the major classes, which is hydrocarbon monoterpene. And then for cone, the main compound in cone oil was B-pinene, 33%, and we are also alpha-pinene, 29, and beta-myrcene, 4%. Our results are in great agreement with the work of Mascioni and al., who reported alpha-pinene as the main compound, the difference part of P-pinister essential oil growing in central Italy. However, there are some difference in the amount of major compound. In fact, various factors could influence essential oil composition, including genetic, biotic, and abiotic factors. Now we will see the herbicidal effect. On Petri dishes, seeds were put on double-layered Whatman number 1 filter paper containing different concentration, which is 1, 2, and 3 microliter per ml of essential oil dissolved in 1% between 20 solution. Then every culture was incubated under controlled condition. The tested read were Sinapis arvensis and Fullerenes canariensis and Triticum durum as a crop. The evaluation considered the germination, the shoot length, and the root length. Germination and seedling growth were strongly inhibited under essential oil treatment in a rate-dependent way, and all weeds tested display different degree of sensitivity toward the essential oil. In addition, Sinapis arvensis was more sensitive to all tested oil than Fullerenes canariensis, and Triticum durum was the most resistant. As we can see in the graph, for the germination, we can see that the Fullerenes canariensis showed a moderate inhibition against the 3 essential oil, while Sinapis arvensis showed a complete inhibition with the essential oil of needles and cone. Well, for Reticulum durum, it was very resistant. For the root length, we can see always that Reticulum durum was the most resistant and Sinapis arvensis was the most sensitive. And the same thing is observed for the shoot length. Well, for the Sinapis arvensis, at the dose of 2 microliter per milliliter essential oil extracted from cones and needles ensure the complete inhibition on the germination, the first graph, and seedling growth. We see always that at the highest concentration, 3 microliter per milliliter, there is always complete inhibition, especially for Sinapis arvensis. And for Fullerenes canariensis, appears to be slightly resistant at the same dose to the 3 tested essential oil, which is clear in the 3 graphs. Well, for Reticulum duratum, there is a low effect was obtained with branches oil when compared with needles and coiled essential oil, which is here. We can see that at the highest concentration, we haven't seen any complete inhibition. Obviously, herbicidal activity was varied over dose, tested oil, and tested wheat. If we compare the effect of each essential oil, it may be concluded that the essential oil from cones and needles possessed more herbicidal effect than branches essential oil. Now we will see the antifungal effect. The 5 milliliter in diameter of mycelia disc extracted from the periphery of a 6-day-old culture was inoculated of each pedia plate and finally incubated in the dark at 24 degrees. There is 5 tested fungi: Fusarium Colmeram, Fusarium graminarium, and Fusarium avinaceum, Bipolaris soriciniana, and Botrytis cinerea, which are very harmful again and attack the cereal production in Tunisia. So, in fact, the test of antifungal activity showed that oil obtained from needles, cone, and branches significantly reduce the growth of all the tested fungi, which is clear in the graph. We can see here that Fusarium graminarium was the most sensitive, while the Bipolaris soriciniana was the most resistant against oil extraction from cone, branches, and needles. Essential oil extracted from the 3 parts of Pampinester essential oil showed a different effect. The lowest rate of the lowest rate of inhibition was observed when applying essential oil extracted from branches, which is clear here, where the most important effect was with needles. Indeed, 4 microliter per ml of essential oil from cones ensure an inhibition of 40% of the fungal species tested. As a conclusion, essential oil obtained by hydrodistillation from the 3 aerial parts of Penis maritime were mainly composed of hydrocarbon monoterpene and have shown a promising activity, a strong phytotoxic and antifungal effect that may represent useful and valuable leads for the development of a new pesticide. Based on our preliminary results, the essential oil of cone and needles could be suggested as alternative herbicide and fungicide. However, for a better and optimized formulation of a potential biopesticide, further studies are required to determine the biological standardization, quality control, cost, applicability, safety, and phytotoxicity against cultivated crops. Thank you for your attention.
Thank you, Dr. Sana. Thank you for your detailed presentation.
Thank you.
To Dr. Sana, is there any question in the group? Okay, thank you again.
Thank you.
Have a good day. Thank you. The next one, determination of the anti-inflammatory activity of nanospray-dried nanoparticles within in vivo HETCAM, a study that can be used in postoperative wound healing. Dr. Tilbe Çevikelli.
Thank you. Is there any problem with the voice or the presentation now?
Thank you. Welcome.
Is the voice okay and the presentation, can you see it?
Yes, okay, no problem. It's okay.
Thank you. I'm Tilbe Çevikelli from Çukurova University, Department of Pharmaceutical Technology. I'm undergraduate student. And our study is titled Determination of Anti-Inflammatory Activity of Nanospray-Dried Nanoparticles with in vivo heat chemistry that can be used in postoperative wound healing. Ketorolac-tormetamine is a non-steroidal anti-inflammatory drug, acts as a non-selective COX inhibitor, and it's often used successfully in post-surgical and pain and surgical wounds. Wound healing is a process of tissue regeneration that includes inflammation as an early step, whereas anti-inflammation is a positive response to treatment. The chronic nature of non-healing wounds prompt the development of nanotechnology-based therapies aimed at speeding up the healing process and mending the damaged tissue. Spray drying is a fast and simple technology. The key advantage of nanospray drying is the production of drug-loaded nanosized particles for various drug delivery applications. The in vivo heat chemistry is very sensitive to both steroidal and non-steroidal agents and well established for screening anti-inflammatory effects of therapeutics and drug formulations. Within the scope of this study, ketorolac-loaded nanoparticle-based drug delivery systems, which can be effective in pain treatment and wound healing processes in postoperative conditions, were prepared. Then in vivo heat chemistry was performed on the selected optimal formulation for anti-inflammatory activity. As material and methods, nanoparticles were prepared using nanospray dryer butyl. Erdogid RS100 was used as polymer, and nanoparticles were characterized as particle size, polydispersity index, zeta potential, entrapment efficiency, and dissolution kinetics. The inlet temperature was 100%, the outlet temperature was 54 degrees, pump level was 4, and spraying level was 100%. For in vivo anti-inflammatory activity by heat chemistry, for preparation of the test samples, ketorolac-loaded nanoparticles equivalent to 100 micrograms per pellet, and pure ketorolac, again 100 microgram per pellet, and SDS were suspended and dissolved in an agarose solution. For ease of application, these solutions were cooled to room temperature for solidification and applied on the chem tissues. The in vivo heat chemistry was conducted as the previously incubated cells were positioned horizontally and rotated several times. The eggs were opened on the snap site and 10 to 15 milliliters of albumin were aspirated from them. The eggs were traced, and after that shells were removed with the forceps. The cavity was covered with the film and they were incubated for further 72 hours. Pellets were placed on the cam, which had approximately a diameter of 2 centimeters. One further day later, eggs were evaluated under the stereo microscope. A total of 10 to 15 eggs were utilized for each test compound, and also the experiment was repeated 3 times for each test compound. In figure 1, here you can see the evaluation of heat chemistry. And for statistical and mathematical analysis, ANOVA test was used, and results were each represented as mean and ST values. The semi-quantitative score system was used for evaluation of chem activity, which can be seen in table 1. In table 2, you can see the score index in the proportional inhibition of the inflammation, which is the evaluation graph for us. For results, The in vivo anti-inflammatory activity results indicated that the nanoparticle formulation, including ketorolac as 100 milligrams per pellet, shows significantly strong anti-inflammatory potential, approximately around 85% inhibition value and no embryotoxicity, whereas ketorolac itself showed weak activity around 60% inhibition on the chem. In figure 2, you can see the in vivo anti-inflammatory effects of ketorolac on graph A and ketorolac-loaded nanoparticles on the graph B on the chem. As discussion, the anti-inflammatory activity of ketorolac-loaded nanoparticle formulations was examined using the in vivo heat chem assay. For the assay, STS was used to cause irritation hemorrhage on the chem. In line with the heat chem results, it is thought that the anti-inflammatory activity has been increased with the nanoparticle structure, and thus a successful treatment can be obtained in the wound healing process. Also, similar results are found in literature. In conclusion, ketorolac-loaded nanoparticles were produced in nanospray drying device. With the obtained heat chem results, it has a hope of successful nanotopical formulation, especially in wound healing. These are our references, and thank you for your kind attention.
Thank you, Dr. Tilbe Çevikelli. Thank you very much. Is there any question to Dr. Tilbe? Okay, thank you very much. Now the next turn. Magdalena Rostkowska, effect of diet supplemented with cadmium.
Hello, everybody.
Hello.
It's
Welcome.
a pleasure for me to see all of you here during this Congress. I hope that everything is okay with my internet. Can you hear me and see me? Because I'm not sure. Yeah?
Yes, we can see you. You're welcome.
Yeah. Okay, thank you very much. You can't see here my name. Here is the name of my friend Dagmara, because I'm on my winter holidays. And I haven't got my computer with me,
problem. No
so
problem.
I had to land this one from my friends, but it's me.
Okay.
So I will present you my presentation from the beginning. Ladies and gentlemen, I wanted to present you the results of our studies, which are connected with the diet supplemented with cadmium and the effects of this diet on cell death in the soil invertebrate Ditobius forficatus. As we know, there are many stressors which can affect the animals, plants, and humans' organism. Such stressors are, for example, pathogens, electromagnetic field, radiation, lack of water, starvation, but also many xenobiotics, which are, for example, heavy metals, toxins, pesticides, and all such stressors can cause the death of cells, of tissues, organs, and eventually the entire organism. We know numerous types of cell death. However, only 2 types are well known: autophagy, apoptosis, and necrosis. While apoptosis and necrosis are types of the typical cell death, autophagy, depending on stressor, on the strength of the stressor, it can be a survival pathway or can be a cell death. During our studies, we are interested in activation of all these 3 processes. And of course, cadmium, it's a heavy metal, which is present in natural polluted and non-polluted environment, and it origins from natural processes as well as from anthropogenic activity. Cadmium can accumulate in many organisms, and after eating them, cadmium can be transferred to the higher levels of the trophic chain. So it is very important to analyze how cadmium can accumulate in one organism, and after eating it can be accumulated in another one, and what is the effect of this heavy metal. Thus, the aims of our studies were to investigate and analyze and describe all alteration caused by cadmium short and long-term intoxication at different levels in the organism. So I mean from the level of tissues, cells and organelles, and also some enzymes which are produced by many organelles. The second one is the analysis of activation of cell death, apoptosis, and autophagy after cadmium supplementation, and to state if the digestive epithelium fulfills the role of a barrier against heavy metal effects. We were interested in the soil centipede, Litobius forficatus. It is the common European species which lives in the soil, and it can feed with many other small invertebrates. So during our experiment, we collected adult specimens of Litobius in the southern Poland, and after acclimatization to the laboratory conditions, Litobius were divided into 3 groups: the control group, the CD12, and CD45. And now I would like to explain you what are the differences between these 3 experimental groups. Animals from the control groups were fed with Chironomus larvae. They are larvae which lives in a freshwater environment, so they were maintained in a clean water for 7 days. Animals, Litobius forficatus, from CD12 groups were fed with Chironomus larvae, which were maintained in water, supplemented with 80 milligrams per liter of cadmium chloride for 7 days. And such group of Litobius forficatus were fed with this Chironomus larvae for 12 days. And the last experimental group, the same conditions, but it was the long-term experiment, so 45 days. After these experiments, we isolated 4 organs: midgut, salivary glands, ovary, and testes. However, here I would like to show you only the small fragment of the results of our studies, which are connected with the midgut, mainly the midgut epithelium. And we prepared all these organs for the confocal microscopy, flow cytometry, and transmission electron microscopy. The midgut, the part of the middle part of the digestive system, is lined by the epithelium, and this epithelium is composed of the digestive cells, the secretory cells, and regenerative ones. It lines on the basal lamina, which is surrounded by the visceral muscles. However, from the food masses, it is separated by the peritrophic membrane. So we were interested in just this midgut epithelium because it is the kind of barrier against all stressors which can attack the organism from food masses. During our studies, we wanted to analyze how reactive oxygen species are produced. So we used the confocal microscope and the flow cytometry. And here I wanted to present you this part connected with the midgut here and this one. So you can see that after the prolongation of cadmium feeding, the number of cells which were ROS positive were increasing, distinctly increasing. So after 12 days and 40 days, here we can also see the results from the flow cytometry. The transmission electron microscope showed also different changes. After 12 days of cadmium intoxication, we observed that numerous vacuoles with electrolucent content appeared, many organelles were destroyed, and the process of autophagy appeared. So organelles were digested inside. Additionally, we observed that the number of storage material decreased. We observed only glycogen granules and lipid droplets, while proteins and polysaccharides different than glycogen were not present. After 45 days of the experiment, we observed strong necrosis. So the cytoplasm was poor in organelles, it was electrolucent, numerous vacuoles appeared. However, small amounts of glycogen granules and lipid droplets were also observed. Because we are interested in organelles, we wanted to check how mitochondria were changing. Because as we know, they are very, very important organelles which take part in cell death processes. So we also, the transmission electron microscope showed the really terrible changes in all these organelles in this mitochondria. So we can see that the number of the crystals were decreasing, and some small vacuoles were appearing inside. So the flow cytometry revealed also the increase of the number of mitochondria with a changed membranous potential, what means that such mitochondria take part in the cell death. And eventually the apoptosis, we also observed that after the prolongation time of the cadmium intoxication, we also observed that in confocal microscope that after 12 days of the experiment, the number of apoptotic cells increased. So you can see here such pink spots. It is the tunnel assay. After 45 days, the number of apoptotic cells decreased slightly. So here we can also see, here is the number of early apoptotic cells after 12 days. Here is the number of after 12 days, the number of late apoptotic cells after 12 days and after 45 days, and the necrosis. So the flow cytometry also confirmed such statement that after 45 days, the processes of necrosis were activated. So what are the conclusions of our studies? Short-term intoxication causes intensification of autophagy and digestion of reserve material. Long-term exposure to cadmium causes inhibition of autophagy connected with the lack of reserve material and the activation of cell death processes. Autophagy and cell death are nutrient deprivation induced processes. It is really very interesting. And digestive epithelium fulfills a role of a barrier against cadmium effects. Thank you very much. I wanted to say that the study was financed by the Natural Science Center in Poland. So thank you very much for the attention, and it was really a great pleasure for me to attend this Congress. Thank you very much.
Yes, thank you very much. Thank you very much. The next, Dr. Sinan İnce, the protective effect of boron. Yes.
Can you see? Okay. Thank you very much. Our study, firstly, our study is PhD thesis for first author, and this study was supported by Afyonkarahisar University Scientific Research Council. And the title is the protective effect of boron agents aflatoxin B1 induced liver injury in male rats. Aflatoxins are the most potent naturally occurring hepatic carcinogens. Because of their prevalence and toxicity, aflatoxins are of paramount importance to public health worldwide. Aflatoxins cause increased activity of liver enzymes in serum, and aflatoxin B1 damage cells and DNA by inducing active oxygen species, causing oxidative stress as well as genetic changes. that lead to DNA damage and mitochondrial permeability changes. It's stated that aflatoxin B1 increases the expression of apoptotic genes PX and caspase-3 in the liver and causes a significant decrease in BSL2 expression, thus inducing apoptosis via the mitochondrial pathway. It has also been reported that aflatoxin B1 increases the expression of the NFKB gene and protein. Aflatoxins are immunotoxic and reduce natural resistance to disease and impair vaccines-induced immunity. Boron is a mineral that has vital roles in plant, animal, and human metabolism, and it has an important role in osteogenesis and osteoarthritis. Boron positively affects the use of sex hormones such as estrogen, testosterone, and vitamin D. Boron reduces TNF alpha and inflammatory biomarker levels, and it also increases the scavenging of free radicals by increasing the level of antioxidant enzymes such as peroxidase, catalase, and glutathione peroxidase. In this study, it was aimed to determine the possible protective effects of boron agents liver damage in male rats given aflatoxin B1 in vivo for the first time. For this purpose, some biochemical parameters, lipid peroxidation, and glutathione levels activity determination of antioxidant enzymes and DNA damage were determined. In addition, mRNA expression levels of apoptotic and inflammatory stakins in liver tissue and histopathological examination of liver tissue were performed. A total 30 male spermatozoids were used in the study. These animals were obtained at University, and the selected dose of aflatoxin B1 and boron were determined by examining previous studies. The experimental phase was carried out within 21 days. And 6 groups, control and control groups, the other groups in the aflatoxin B1 group and the other different dose of boron were given to rats. In the results, lipid hepatic liver enzymes of SAT, ALT, and ALP levels was increased in aflatoxin groups, and in a dose-dependent manner, boron treatment decreased these levels. And MDA levels in aflatoxin group, these levels were increased in liver and blood, and boron treatment decreased these levels. And glutathione levels in aflatoxin Weber in the group were decreased, and boron treatments were increased these levels. Supraoxidative dismutase and catalase activities were decreased in aflatoxin groups, and boron treatment in a dose-dependent manner were increased these levels. In the apoptotic gene expressions, BX, caspase-3, caspase-3, 8, and P53 levels were increased in the aflatoxin group, whereas boron treatment in a dose-dependent manner were decreased in these levels. Also, anti-apoptotic gens BCL2 levels were found to be low level in the aflatoxin group, and in boron treatment, treatments were increased this level. TNF alpha and NFKB levels, which are inflammatory genes, were increased in aflatoxin group, and boron treatment decreased these levels. And also ssDNA damage were increased in aflatoxin group, at the same boron treatments decreased these levels. Histopathological evaluation in the aflatoxin groups in liver, degenerative changes in hepatocyte in the pericentral region, biomarker structure deterioration, mononuclear cell infiltration in the pericentral regions, and the nucleate hepatocytes were shown in the aflatoxin groups, and boron treatments in a dose-dependent manner improved this hepatic injury. In this study, it was determined that liver enzyme activities, lipid peroxidation, DNA breaks, pro-inflammatory and apoptotic gain expression levels increased, glutathione and antioxidant enzyme activities decreased, and histopathological changes related to the liver tissue damage occurred in last given aflatoxin B1. It was determined that boron applications at dose of different concentration in a dose dependent given together with aflatoxin B1 deposed these negative changes. As a result, it was revealed that boron exhibited protective effects against aflatoxin B1 induced liver damage with antioxidant, anti-inflammatory, and anti-apoptotic effects. Thanks for listening.
Thank you, Dr. Sinan. Is there any question for this presentation, for this study? Thank you, Dr. Sinan.
Thank you very much.
You're welcome. The next one, Dr. Seda Beyaz, the investigation of therapeutic activity of Fullerenes P60 against liver tissue damage by biochemical and histopathological analysis. Dr. Seda Beyaz.
Hi, everybody. Welcome to Second International Congress on Biological and Health Science. I am Dr. Seda. My work that I will present to you, investigation of therapeutic activity of Fullerenes 60 against liver tissue damage by biochemical and histopathological analysis. The liver is an important metabolic organ in the body, secretes and various nutrient protein. Most importantly, the liver function and the detoxify tissue by neutralizing and elimination toxin through hepatotoxic mediated enzymatic detoxify system. It has been determined that DMBA is associated with the development of breast carcinoma, high level of tumorigenicity and oxidative stress marker, in carrying DMBA-induced memory carcinoma, the production of reactive oxygen space and membrane lipids show a significant increase in tumors due to peroxidation. Fullerenes 60 is an aromatic and spherical molecule that belongs to class of carbon molecule and consists of 60 carbon atom. Fullerenes 60 is a spherical molecule in nanocarbon structure containing double bonds. Fullerenes 60 has a potential anti-cancer activity and has been that its derivatives exhibit potent immunomodulatory pro-inflammatory activity. Lipid peroxidation, malondialite analysis in liver tissue, catalase activity, glutathione and total protein level were determined by spectrophotometer. In addition, liver tissue were evaluated by histopathological material at method, animal material and research group. The animal experimentation section of this study from University, it was conducted in the Experiment Animal Research Center. Experimental group, control Fullerenes 60 group, DMBA group, Fullerenes 60 and DMBA group. Application control group, the group fit a standard yet but not given Fullerenes 60 and DMBA. Fullerenes 60 group fit standard yet not given DMBA but given Fullerenes 60. DMBA group application, the group fit a standard yet and given DMBA. Fullerenes 60 and DMBA group application group given Fullerenes 60 and DMBA. Preparation and application of DMBA. 45 milligram kilogram DMBA was dissolved in 0.5 milliliter sunflower oil. The administration of DMBA was carried out by injected 45 milligram kilogram live weight together with sunflower as oral device start from the first day when the rats were divided into 4 groups so that their live weight were close to each other. One a week for 3 weeks. Preparation and application of Fullerenes 60 0.78 milligram milliliter Fullerenes 60 dissolved in 1 milliliter olive oil. Fullerenes 60 taken from the stock solution was administered to animals at dose of 1.7 milligram kilogram body weight 5 times a week as an oral device start from the second week and the study continued for 16 a week. Liver tissue malondialite measurement were carried out in spectrophotometer at a wavelength of 522 nanometers. liver tissue glutathione measurement were carried out in spectrophotometer at a wavelength is of 412 nanometer. Liver tissue catalase activity measurement were carried out in spectrophotometer at a wavelength of 240 nanometer. Liver tissue histopathological analysis at the end of research section from liver tissue will be fixed in 4% formaldehyde solution and examined under a light microscope. Statistical analysis, all data were evaluated with analysis of variance in SPSS 22 PEC program, one-way ANOVA post-hoc Seda test, and were application to determine the difference with graph. Result, liver tissue malondialdate measurement result, liver tissue malondialdate level in the DMBA treated group has increased because an increase in oxidative damage in the control and treat Fullerenes 60 has decreased the malondialdate levels. Liver tissue glutathione measurement result, when we looked at liver tissue glutathione level, it was found that there was an increase in the group treated with Fullerenes 60 and decrease in the group treated with DMBA. Liver tissue catalase activity measurement result, when we examined liver tissue catalase activity, it was found that, there was an increase in the group treated with Fullerenes 60 and decrease in the group treated with DMBA. Liver tissue histopathological analysis results control group. Since there was no damage in the control group, no inflammatory cells were observed. In the Fullerenes 60 group, the black arrow indicates the central vein. The portion of DMB indicated by black arrow show congestion expansion, focal necrosis, and inflammatory cell formation in the sinusoid. Fullerenes 60 and DMBA group portion indicate by black arrow show congestion, inflammatory cell, and hydropic degeneration in the sinusoid. Conclusion, for this region, Fullerenes 60, which has been more important in recent study and draw attention with its many biological activity and various molecular targets, is important in order to contribute to treatment method, especially with its protective and preventive effect on liver tissue damage. Reference. Thank you for listening.
Thank you, Dr. Seda Beyaz. Is there any question for Dr. Seda's presentation? Okay, thank you, Dr. Seda. The next one, the relation of protective effect of Fullerenes. Sorry. Dr. Özlem Gök. Yes.
Hello, my dear teachers. My working title is like this: Relation of protective effect of Fullerenes 60 nanoparticle against lung tissue damage in rats with biochemical and histopathological biomarker. Nanomaterials are widely used in many fields from technology and industry to medicine. Considering its physiochemical and biochemical properties, it has wide uses. Fullerenes 60 is a nanomaterial consisting of 60 carbon atoms. Fullerenes 60 discovered experimentally in 1985 is the most abundant and responsive of the stable Fullerenes ever isolate. One of its most biological important properties is that it is a redox activity molecule that can reduce various reactive oxygen species. In addition, Fullerenes 60 has been reported to indicate inflammation, immunotoxicity, and respiratory toxicity in rodents. The DMBA we used in our study is a polycyclic aromatic hydrocarbon format drying in incomplete combustion of carbon compounds found in tobacco smoke and motor vehicle exhaust emission. Lipid peroxidation, malondialdehyde analysis in lung tissue, catalase activity, glutathione, and total protein levels were determined by spectrophotometer. Lung tissue was examined histopathological. Animal experiments section of this study, Pratt University with the permission of the Animal Experiments Ethics Committee. In this study, 60 family rats were used. Rats 12 hours of light per day. A lighting period of 12 hours was a period. Groups, control groups, C60 groups, DMB groups, C60 and DMB group. Control group, the group fit a standard diet but not given C60 and DMB. C60 group, C60 1.7 milligram kilogram body weight oral gavage. DMB group, DMB 45 milligram kilogram body weight oral gavage. C60 and DMB group given C60 1.7 milligram kilogram body weight oral gavage and DMB 45 milligram kilogram body weight oral gavage. Trial period 60 week. Lung tissue, malondialdehyde, glutathione, catalase activity, and total protein density measurements. After adding the necessary materials, malondialdehyde measurements were measured in spectrophotometer and a value of 532 nanometer. Glutathione 412 nanometer. Catalase activity 214 nanometer. And total protein density was measurement and 650 nanometer wavelength in a spectrophotometer using library protein ESI kit. Histopathological studies. After the rats were decapitated, the lung tissues of each rat were removed. A portion of the lung tissue was fixed in formaldehyde solution and embedded paraffin. Then lung tissue samples were stained in section with hematoxylin and ozone. The prepared section were examined under the microscope. All data were evaluated with analysis of variants in SPS 22-page program. Lung tissue malondialdehyde measurement results. Compared to DMB group, lung tissue malondialdehyde levels were significantly reduced. C60 treatment group. There was no statistical control and difference between the control and C60 groups. Lung tissue glutathione measurement results. There was no statistical significant difference between the control and C60 groups. It was determined that the glutathione level increased in the treatment group given C60 and DMB, and highest glutathione levels were in the control and C60 group. Lung tissue catalase activity measurement results similar to glutathione levels. Histopathological results. Control groups, normal histology observed in the control groups. C60 group, normal histology was observed in the C60 groups. DMB group, severe edema formation was observed in the DMB group in terms of increased thickness of the interalveolar wall and inflammatory self-infiltration. Significant damage was observed in the DMB injury group. C60 and DMB group, there were a reduction in interalveolar wall thickness, inflammatory cell formation, and hemorrhage in the C60 and DMB treat group compared to the DMB treat injury group. These results show that Fullerenes C60 protects the lung tissue from the toxicity of DMB and reduces in the damaged lung result of toxicity, and may contribute to new treatment protocols to be developed in future studies. In conclusion, we can say that Fullerenes C60, which has antioxidant and anti-inflammatory properties, has protective effect on lung tissue and can be used against lung tissue damage. Thank you for listening to me. Hocam sesiniz gelmiyor.
Thank you, Dr. Özlem. It's very good presentation and very fast. Thank you very much. Now we finished this section. First, I want to ask in the program, is there, sorry, Nur El Huda Bekar. Is she here?
Ali, ben bir ses açayım.
Evet hocam. Nur El Huda Bekar ve Anjali Ranjan. 9 başlıklar. Onlar
Hocam kontak kurmaya çalıştık. Ulaşılmıyor.
Tamam. Okay, thank you very much to all participants. In this section, it will finish. We come to at the end of this section. Thank you very much again, all participants, all presentations. Thank you.
Dear Professor Dr. Kadir Öymen Hançerlioğlu, we appreciate for your support and for your moderation. It was very nice to do something together, and thank you very much for your presence again.
Thank you.
Dear respected participants, we will continue in 15 minutes with the section of invited speakers. Meanwhile, we will put the poster presentations with the music. Enjoy the music, please, and see you in 15 minutes again.
Thank you very much.
See you. Dear respected participants, welcome to our invited speaker sections. Today, we have got very interesting topics like yesterday, and first presentation is going to be held by Professor Dr. Ardo Sabir from Indonesia, from Hasanuddin University. Dear Professor Dr. Ardo Sabir, we would like to happy to listen you.
Thank you. Thank you, Dr. Ulas. Can you hear my voice?
We are not able to see your video. Now we can see you.
Okay. Can I present my slide now?
Yes.
Okay, thank you very much. First, good morning in Turkey and good afternoon in Indonesia. Assalamualaikum warahmatullahi wabarakatuh. I'm very happy to be here. Although we meet in online by Zoom. Thank you for organizing committee of Congress, especially for Dr. Ulas, who invite me to be a speaker, invite speaker in this Congress. Okay, to efficient the time, I will start my presentation. Can you see my slide?
Yes, we can see.
Okay, I hope it's not trouble. Okay. In this time, I want to present my my title is the potential role of propolis in relief dental pain, or people commonly said toothache. Okay, you can see my slide.
Yes, we can see your slide, but it is not full screen.
This is my outline, introduction, dental pain, propolis, the role and mechanism propolis in relief dental pain of toothache, and the last one is conclusion. Okay, we going to introduction. Okay, the dental pulp, as we know, is the connective tissue uniquely situated within the rigid eschasement of remineralized, we call dentin. The inflammatory response to dental.
Dear Professor, sorry for interrupting, but it is not full screen, and it doesn't change. It is on the first one. Yeah, now it's changing.
Okay, I think maybe this is trouble with if I full screen. So can I present with this?
Maybe we can do it so. You can send the presentation to me by WhatsApp, and I can open from this computer and I can change. If it's not easier for you in this way.
Can I present with this pop?
And now it doesn't change. Now we are seeing introduction section.
You can see my slide introduction. This.
Now the application of propolis in dentistry, it is written.
Yes. So the slide is moved. Okay.
Yeah. If you can change this slide order, then it is also visible. No problem.
Yeah, there. Push there, Professor Sabir. Push two times. 2 times.
2 times.
Yes. So, or longer.
This
Yes,
icon.
yes, yes.
You can see my
Okay.
slide.
If it's not working, you can close the PowerPoint and share screen and start again the share screen,
Okay.
but go to
Okay, I will try.
push two times on share screen. Share screen and then again share screen and then open the PowerPoint. Yes,
This?
now it will work. Yes. And now go to the first slide. Go up to the first slide to start from the beginning. Yes. Yes. Double click and then go to the down to that special button.
This one?
Yes, yes, that one. Yes. Click long. And wait a bit.
You can see?
A bit.
Not yet?
If not, then go up to the slideshow. Up above on the left. Yes, above, more up. Yes, slideshow. Yeah, try there.
From beginning?
Yes. Try there. Click.
Okay, already. Yes, I click it.
Okay.
Can you see?
Oh, yeah. It's not working. Yeah.
Not working.
Not problem. We can see you also your
Yes.
presentation. The main problem is every time you have to click on the next slide to change the slides.
Okay, you can see this is introduction. No?
Yeah. Yeah.
Yes. Go slide after slide. Slide after slide. It's okay. It's okay. It's a bit smaller, but it's okay. We can see.
You can see now?
Yes.
The application of Propolis in dentistry?
Yes.
Okay, okay. Okay, I can start?
Yeah.
By introduction, okay?
Yes, yes. Do it. Do it. Good luck. Good luck.
Okay, okay. Okay, thank you. Maybe it's slow my program. I'm so sorry.
No problem.
Okay, as introduction, as we know, the dental pulp is the connective tissue uniquely situated with the rigid encasement of mineralized we call dentin. The inflammatory response to dental injury to dental pulp has a major significant clinical significance. The injury may be caused by dental caries, commonly is dental caries, the bacteria, dental restorative procedure, tooth fractures, and also attrition by the mastication. Data from my country, research, basic health research by our Minister of Health in 2018 show that the prevalence dental pain in Indonesian population is 57.6% in our population. So the use of the natural product and holistic or alternative medicine has popular among our society because the potential side effect and safety concern of conventional allopathic formulation. As we know, as the propolis is a natural product derived from plant resin collected by honeybee and also already used by in folk medicine for centuries. Okay,
change the slides. Change the slides, Professor Sabir. Change the slides.
Yes, I'm already changed. Can you see?
No, it doesn't change.
Dear Professor, if you can send your presentation to me, I can share it from here and I can change it for you.
Yes, it is a good idea. Yeah, now it changed. Now, how did you do it? Do it again, change again. Yes, now it's working.
Okay, can I present by this form? Not full screen?
Full screen is not the problem. The problem is during your speech, the slides are not changing. You have to do it manually or I can do it for you.
No, he's doing now. He's doing now, Ulash. He's doing a change. Now we see introduction. Now go to the next one. Yeah, we see now about this graphic with pedodontics and so on. Okay. Now, now it's changing. Aim of presentation. Now it's okay. You can change.
Okay. The application of propolis in dentistry, many fields of dentistry has been used propolis in their treatment, like oral surgery, oral medicine, oral pathology, pedodontics, periodontology, and also conservative dentistry and endodontics. So the aim of this presentation is to highlight the potential role of propolis in relief dental pain of toothache caused by dental pulp inflammation. This is dental pain. This is briefly about dental pulp. Dental pulp is the part in the center of tooth made up of living connective tissue. It contains several components like cells. Major is the fibroblasts and undifferentiated mesenchymal cell and other cells. The second is fibrous matrix, like collagen fibers type 1 and type 2. And the last one is the ground substance. It's environment surrounding the cells and fibers of pulp, and rich in proteoglycans, glycoproteins, and also water. Okay, this is about dental pain. Dental pain, we know that the dental pulp is abundantly innervated by the both sensory as anomics nerve fibers. The nerve fibers enter the pulp through apical foramen along with the blood vessels. International Association for the Study of Pain define the dental pain is as an unpleasant sensory and emotional experience associated with the actual or potential tissue damage, or describes in terms of such damage. Dental pain is usually caused by either noxious physical stimuli or the release of the inflammatory mediators that stimulate receptor located on the terminal ending of nociceptive afferent nerve fibers. The activation of the dental pulp nerves by this physiologic, like thermal, mechanical, and chemical stimuli result in a pure sensation of pain. The mechanism of inflammation. Inflammation is a complex biological response of vascular tissue and immune cell to harmful stimuli, like to the instant irritant, damaged cell, and pathogen, which can cause severe pain. Can be present by the immigration phagocyte accumulation, the neutrophils, monocyte, macrophages, and lost the tissue function. During the process of inflammation, macrophage stimulate the translocation of nuclear factors kappa beta and activated pro-inflammatory cytokines. Besides that, the activation platelets also stimulate lead the release of arachidonic acid. During the inflammatory response, the lipoxygenase enzyme convert arachidonic acid to the leukotriene and A4 and produce leukotrienes B4. Another side, the cyclooxygenase 1 and OXO2 convert arachidonic acid to prostaglandin H2 and produce thromboxane, prostaglandin, mainly the prostaglandin A2 and prostacyclin, and it leads to the inflammation and also pain. In this figure 1, we can see that how two mechanisms of the peripheral stimulation. of nociceptive nerve fibers of dental pulp. This when the acute pain occur, according to the hydrodynamic theory, that's the stimulate that cause fluid movement, the fast movement of fluid in the exposed dental tubules result in stimulation nociceptive nerve fibers. And when the pain caused by inflammation, this is the release of many mediators of inflammation, including kinins, bradykinins, substance P, prostaglandins, and other mediators inflammation. And also this is we can see the positive feedback loop. So the inflammation to persist by far for beyond cessation of the dental producer. Okay, we we talk about the propolis. The propolis is a natural resin hive product collected by honeybee from leaves and buds of the different plant source. The function of propolis is to preserve warmth in hive and keep out microbes, and also has been used to healing and a part of apitherapy. In general, the raw propolis contain about the mainly resin and vegetables balsam, waxes, essential and aromatic oils, pollen, and also organic materials. The chemical composition propolis is very complex, depends on the collecting time, location, and also the plant's source. This is the pharmacological activities of propolis. We already know, antimicrobial, anti-inflammatory, antioxidant, anti-tumor, regenerative tissue, and also immunoregulatory. This is about the role and mechanism of propolis in relief dental pain. Okay, the major component of propolis is, is flavonoids and also caffeic acid, phenylethyl ester. So this is the biologically active compound. They have anti-inflammatory and antioxidant properties. My research show that the propolis could delay TNF alpha and also the COX-2 expression in dental inflamed dental pulp tissue. And also this is anti-inflammatory in propolis can be explained by the present active flavonoids and cinnamic acid. Study by Bachia et al. Found also that the cafe, cinnamic acid, and also comeric acid in propolis may be involved in the action of propolis to inhibit the production of interleukin 6 and 1 beta by macrophages. They also observed that the cafe and cinnamic acid are strong lipoxygenase inhibitors, suppressing production of leukotriene by peritoneal macrophages. Many research also show that the cafe inhibits the release of inflammatory cytokines and simultaneously increases the production of anti-inflammatory cytokines like interleukin 10 and interleukin 4. The research also showed that the cafe decrease the infiltration of the inflammatory cells like neutrophils and also monocytes. Due to the lipophilic nature is easy to cafe to enter the cells. The cafe inhibits lipoxygenase and cyclooxygenase enzymes, and they involve in the metabolism of arachidonic acid pathway. Therefore, the metabolism is arrest and no release the prostaglandin and leukotriene, which responsible to inflammation and pain. Okay, this is the role. The figure 2 show the role of propolis and its component toward phospholipid metabolism and production of arachidonic acid pathway of the inflammation. So the pathway of the active substance in propolis is different. Like Kp inhibits oxygenase, so the vasoconstriction and production of leukotriene is decreased. The other side, the flavonoids also Kp inhibit cyclooxygenase, so the prostaglandin production is decreased, and mainly the prostaglandin A2. That's conclusion that the propolis as the natural products by honeybee that had been used in apitherapy for a long time due to its various properties. Many studies demonstrate that the propolis and its component has strong inflammatory and antioxidant activities. Thereby could be effective in relief of dental pain of toothaches. This is the reference I used. Okay, this is a little bit about my university, Hasanuddin University in Makassar, about 10 kilometers from center of city. This is Makassar. Okay, thank you very much for your attention. Have a good day.
Dear Professor Dr. Arda Sabir, thank you very much for your valuable presentation. It was very good combination, dentistry and propolis. I think a very low number of people are working in this area. That's why your experience very precious for us. Thank you very much for your valuable contributions. And dear
thank you very much, Dr. Ulas.
Dear respected participants, do you have any questions about the presentation of Professor Dr. Arda Sabir? I'm opening your voice.
Dr. Stephen?
Yes, yes. Congratulations, Professor Sabir, for an excellent presentation. There are indeed millions of people having pains, dental pains all over the world. I noticed one very interesting thing in one of your slides. You said that Kp, caffeic acid phenetyl ester, is lipophilic. This is very good. I did not know that it's lipophilic. It's very good. But now it's all over the world, the people are making the propolis extract in alcohol. And this maybe is not so good for a copy. And we are promoting also the extract of propolis in oils, like in fats. So what do you think? Could you do in Indonesia an oil propolis extract and test on the teeth, on the tooth pain? Maybe it will work better than alcoholic extract. What do you think?
Okay, can I answer the question, Dr. Ulas?
Of course.
Okay.
We are curious about that.
Okay, thank you. Thank you, Dr. Stefan, for your question. It's very important for me, as I know in my knowledge in my country, especially in my university and several universities in Indonesia, who we always join research in about the propolis. Until now, we still use alcohol, methanol to extract the propolis. But I think next time maybe we can try to extract using oils. But I don't think actually I don't know exactly the effect of oil if we used to extract the propolis. But I will try in the future in my experiment and my laboratory to use oil. Can you suggest me what kind of oil we can use to extract, Dr. Stefan?
Yes, there is one method from Lithuania. They presented in 2016, and it is like this. You take raw propolis,
Yes.
like 1 part,
Yeah.
and 4 to 5 parts oil, can be vegetal oil, like your best vegetal oil you have, like sunflower, olive oil, but clean oil. Okay. And then you put in water bath, or you can put also in ultrasounds. There is this special water bath in ultrasounds. And then you increase the temperature. Important is to increase the temperature to 70 degrees Celsius. Okay. Because the oil has not so much power to extract. But if you increase the temperature to 70, and then you can add some polyethylene glycol. Polyethylene glycol. Yes.
Polyethylene glycol.
Yes, and this extract better the active components from propolis.
Okay, thank you very much for the method you give me. I will try in my laboratory to use the oil to extract propolis. Okay, thank you very much, Dr. Stefan.
Thank you very much again for Professor Dr. Ardo Saber. Dear respected participants, we are continuing with the second presentation, which is going to be held by Associated Professor Dr. Zeynep Burçin Gönen from Erciyes University Genome and Stem Cell Center. Her presentation is about the stem cells in oral and maxillofacial surgery. Dear Professor, welcome and thank you very much for your contribution.
Thank you for the kind introduction. I'm trying to share my screen. Is it okay now?
Yes, can see.
Dear colleagues, good afternoon. I am Burçin Gönen from Erciyes University, Turkey. Actually, I'm dentist and oral and maxillofacial surgeon. I am pleased to be here with you. Thank you for the nice invitation, Dr. Ulaş Acaröz, and thank you for the organizing committee. Here is Kayseri, and I actually work in a specific stem cell production laboratory for the patients. It is a good manufacturing practice laboratory. It is GMP laboratory. When you produce the
Hocam
cell...
çok özür dilerim, kesiyorum. Slide'ı ilerletemiyorsunuz herhalde değil mi şu anda? Biz hala ilk sayfayı görüyoruz.
İlerletiyorum aslında.
Şöyle yapalım hocam. Bunu kapatalım, tekrardan açmayı deneyelim mi?
Tamam.
Bir saniye hocam, paylaşmayın. Ben bir ayarı tekrardan güncelleyeceğim. Şu anda. Şimdi deneyebilir misiniz hocam?
Şu an görünüyor mu?
Süper. Thank you very much.
İlerliyor mu? Bir kontrol edelim. İlerlemiyor. Ben İlerledi mi? İkinci slaytta olmamız lazım.
Yok, ikinci slayta ilerle. Hocam yan taraftaki manuel kısımdan ilerletebilir misiniz?
Videolarım çalışıyor mu bu durumda?
Yani öyle manuel ilerletmeniz gerekecek sanırım. Şu anda ikinciyi görüyoruz, üçü göremiyoruz hocam.
Sorry for the technical problem. I don't know why, but you can't see the second slides. And I will do the same. Is it the second slide now?
Now it is the second slide.
Is it third?
No, I think most probably you have to click 2 times on the 3. No. Yes, it is 3 now. Mm-hmm.
Okay, I will try to do it again. It is RGS University Stem Cell Center. I am the production manager of this mesenchymal stem cell unit. We produce umbilical cord, adipose tissue derived from bone marrow and of course from dental pulp, mesenchymal stem cells. And since it is third slide, right? Ulaş, can you see now?
No.
Sorry.
Two.
Okay, I'm just presenting.
Now it's 3. Now
okay.
It's 3.
Okay, since 2013, the lab has produced allergenic and autologous stem cells that have made possible the treatment of over 500 applications in patients with GVHD, diabetic foot, sepsis, and in COVID patients, and in the maxillofacial area, in alveolar bone tissue engineering and temporomandibular joint disease. Actually, if we divide the cell therapies, there should be the somatic cell therapy. You can use stem cells and also the other somatic cells under here. And if you use the cells with any membrane or any 3D scaffolds, we call it tissue engineering. And if you manipulate in gene, it should be a gene therapy. And all these are advanced therapy medicinal products. Here, sorry. And stem cells are one of the most promising tools in regenerative medicine. And in 2081, the discovery of the mouse embryonic stem cells, and after 1998, after the discovery of human embryonic stem cells, the studies becomes faster. And in 2006, Dr. Yamanaka from Japan discovered the human induced pluripotent stem cells, and in 2012 they got the Nobel Prize with this. And in the definition of the stem cells, they are immature and unspecialized cells that have the potential to develop into many different cell lineages via differentiation. And by the conventional definition, these cells can renew themselves, and it is self-renewal. And we have some terms. One is totipotent. Totipotent is the ability to produce a fetus. And the pluripotent stem cells can differentiate any type of the cells. And the potential of the differentiation decrease embryo to adult. And our mesenchymal stem cells are in here. They are in the adult body, and they have the multipotent capacity. The embryonic stem cells are best pluripotent capacity. You can obtain them from the inner cell mass of the 5-day living embryo. However, the ethical, legal, and medical considerations have hindered the development in clinical applications. So we have mesenchymal stem cells. In the adult body, their shape, their morphology is similar. They are fibroblast-like cells. But the International Society for Cellular Therapy has proposed minimal criteria for defining these cells based on their plastic adherence and their phenotype. You can see the lack of the expression of the hematopoietic stem cells marker, and their three lineage differentiation to adipocytes, osteoblasts, and chondrocytes. And in the clinic, they are in the clinic since 2004. And after the first clinical reports, you can find now a very large spectrum of indications in the clinicaltrials.gov. And the important thing is, traditionally, mesenchymal stem cells are isolated primarily from bone marrow and adipose tissue, but in the last clinical trials, the perinatal mesenchymal stem cells and adipose-derived mesenchymal stem cells outnumbered the bone marrow mesenchymal stem cells. This means the non-invasive sources are important. And in the oral and maxillofacial region, we have some non-invasive sources. In 2000, Grontos and friends firstly isolated the dental pulp mesenchymal stem cells, and after then we have several sources from apical papilla, dental follicle, and deciduous dental pulp. And all we know that from these oral and maxillofacial region sources, the developing dental tissues may provide a better source in terms of proliferation. They give a better source for immature stem cells. And in the maxillofacial region, we have temporomandibular joint, and the temporomandibular joint has the own stem cell sources from synovium, synovial fragments, and joint disc. And the other sources may be the Bichat's fat pad, the buccal fat pad, I can say, the nasal polyps and tonsillar biopsy materials has different characteristics and different stem cell sources. From the literature, there are some clinical trials as well. You can see the buccal fat pad here. And from the buccal fat pad, you can isolate the mesenchymal stem cells, and you can gain a large volume of bone. You can see the bone width in the test group. It is higher than the control group. It is one of the clinical trial in the literature. And the cell banking is another issue. And dental pulp, if it is a good source and if it is a non-invasive source, there is an interest on stem cell banking. And I don't use any burr for the pulp isolation because of the cell viability, and I can suggest to crack the tooth for a better dental pulp stem cells. And the advantages of the mesenchymal stem cells to use in clinic is the immune privilege. This means they don't express the HLA class 2 and HLA class 1. And this means they are very useful for the allergenic transplant. You can use without donor match for another patients. And the use of MSCs in the clinic, it requires understanding of the biological characteristics that contribute to therapeutic effects. There is three, we can say. First is the ability to differentiate any type of cells, the differentiation mechanism. And the second is the bioactive secretion. It is the paracrine way. And the third is the immune modulation. Mesenchymal stem cells requires that understanding of these all biological characteristics for the clinic. But in clinic, if we say the cell therapy, we can divide it into two. You can expand the cells, or you can use large volume of the biopsy material, and you don't do any cell culture, and it is minimal manipulation. There is an example for the minimal manipulation is bone marrow aspirate concentrates. And we use these concentrates actually in the temporomandibular joint pain, and it reduced the pain, but there is no microscopic cartilage regeneration because the number of the mesenchymal stem cells are very less. Another source is adipose tissue. Adipose tissue consists of a stromal vascular fraction, SVF, major adipocytes, and extracellular matrix. Normally in Orlan maxillofacial surgery clinic, we use adipose tissue as a microfat and nanofat, the different parts and different emulsification protocols. But if I will show you how it is nanofat and it is microfat, and the filtering systems are different. But if you say it is a cell therapy, this part is cell therapy. These are the components of the adipose tissue, the microfat, nanofat, and after then, if you do a smaller part, these are SVF, stem cells, secretomes, and exosomes, and these are the cell therapy from adipose tissue. And in 2001, Patricia Zook firstly presented the adipose-derived stem cells, and Zook suggested adipose tissue may contain a large, the adipose tissue is available in large quantities, and it is easy to procurement with minimal morbidity and minimal discomfort if you compare it with bone marrow, and it is easy to harvest. And SVF contains adipose tissue stem cells, and if you want to do it with have a large volume of adipose tissue, but if you will expand the stem cells, only 2 or 3 millimeter cube adipose tissue is enough. And for minimal manipulation, you can isolate them with clinical-based devices in operating room. There are several companies in the market, but for the expand the cells, you should have the special laboratory as a good manufacturing practice, GMP laboratories. The most important thing is only 2 to 10% of SVF is mesenchymal stem cells. It is a cell cocktail, and in this cell cocktail, you have another cell sources. I will mention them later. Your cells are in the bottom. The SVF part is in the bottom, the stimescent solution, collagenous solution, natural adipocytes, and it is the oil layer. If you isolate it with mechanical shaking or enzymatic digestion, and these cells, you can see the endothelial cells, muscle cells, adipocytes, and adipose-derived mesenchymal stem cells inside these SVF. And the first step, how we isolate the SVF in our laboratory, sometimes we use the clinical based kits, or we use the standard protocol. First, you should use the Luer-Lok syringes, and first step is removing the blood and tumescent solution, and the second step is the enzymatic digestion, but it is not mandatory. You can just use the mechanical digestion, and after then the centrifugation is the third step. After the centrifugation, we use the filtering filtration is important, and you will have a cell pellet, and you can resuspend this cell pellet with platelet-rich plasma, PRP, or saline, and you can inject wherever you want. I often use these type of cell therapy products in temporomandibular joint pain, and in the literature it is known that you can find the regenerative changes in MRI after using these kind of regenerative products, because we don't have another regenerative option in the cartilage tissue. And you can see the restoration of the meniscus morphology and capsular ligament structure in the MRI. And many efforts have been focused on developing the expansion techniques. The SVF, you don't need to do it under GMP, but if you would like to expand the cells, you should have a special laboratory. This laboratory is in RGS University. It is our laboratory, but in all around the world, in US and in Europe and in all around the world, it is necessary to use the GMP laboratory for expanding the cells. And you don't need very high volume of the tissue. You can use very small dental pulp tissue or very less volume of the adipose tissue for expanding the large therapeutic numbers you can reach in second passage. And it is not for the GMP is not necessary for the stem cell. It is also necessary for the chondrocytes or fibroblasts. If you expand the cells, you should expand in the special laboratory. And another effect of the MSCs is on T cell proliferation. They suppress the T cells, and this means they inhibit the secretion of interferon gamma, and it's very important for immunomodulatory effect. And one of our paper, we use these cells in GVHD, as I mentioned before, and the acute and chronic responses of the patients and the survival of the patients are higher as expected. But the most important thing is they also provide the immune restructuring of the patients. And in oral and maxillofacial region, we use these cells for chronic GVHD because they have the painful oral ulcers in the mouth, and it impairs the feeding function and the quality of the life. And when you infuse these cells from IV infusion, because they cannot open the mouth for a local injection, you can see how it's improved and the ulcers disappearance. And another effect of the mesenchymal stem cells are they promote angiogenesis. And in oral and maxillofacial area, we use this property of the MSCs for the graft survival. Normally, when we use fat grafting, the main complication is the resorption. Approximately 50% of the graft is resorbed. However, when you use the fat graft with MSCs, it affects the resorption of the graft material, and it is very important for the facial area. And another important thing is the migration, because MSCs express a range of chemokine receptors, allowing them to migrate towards the chemokine gradient. And when there is an injury, it is a normal healing mechanism, like SDF1, which may trigger the requirement of MSCs from the bone marrow or from the microcapillary of the tissue. And when you make an infusion to the patients, they will find the way of the injury with these chemokine receptors. And we make a bisphosphonate-related osteonecrosis of the jaw model in a rat, and we applied the cells from tail vein, and we tried to find the fluorescent cells in the mandible. And yes, there are osteoclasts, they differentiated as osteoclasts, and there we find the cells in the mandible, and this is homing effect. This migration capacity is a homing effect, but you can inject the cells by locally. It is our study with orthopedic department. We injected the cells by locally, and local injections may be useful for under specific conditions. And I will show you one hour patient how we see the cells, how we transport the cells to the clinic. It is one of our patients with alveolar defect. Normally we use iliac crest bone grafting, but it is a project of us. We use mesenchymal stem cells from the patient's own wisdom tooth, and this is the cells. We suspend the cells in saline. You should use these cells in 48 hours because they are viable cells, and if you don't use them in 2 days, they will go apoptosis, and it affects the treatment. And you can see we use the platelet-rich fibrin scaffolds as a biological scaffolds, and we inject the cells to this area. That's fine. And lastly, I will talk about exosomes because the new studies are in the literature focus on the exosomes because of the paracrine effect of the cells. They are responsible for the intercellular communications, and they have specific contents such as messenger RNA, microRNA, proteins, and exosomes are internalized by the target cells. And we isolate the exosomes of MSCs, and we have a project with microbiology unit and hematology unit with immunosuppressed patients. We tried the exosomes have the antimicrobial capacity, and we use them as a oral rinse. And you can see the after photo of the patients. Or some another protocols, we use the kit here, the in vitro kit. Oh, sorry. And it is an again temporomandibular joint. It is a rabbit model. The anatomy of the rabbit is a bit different from the human. We use a skin incision, and after then, normally we can reach the temporomandibular joint easily in human, but in the rabbit, we remove the zygomatic process overlying the temporomandibular joint, and we reach the condyle of the mandible here. And after then, we do a model of osteoarthritis, and I will show you the photographs here. Here you can see the zygomatic process, and it is the defect. And we use, this is the mesenchymal stem cell flow cytometer, and this is the exosome quantification. And in the histology, you can see the gap in the control group, and you can see the hyaline cartilage and a good bone regeneration here. And in the statistically, bone and hyaline cartilage regenerations were clearly greater and statistically significant in exosomes group. This means we don't need the cells, the paracrine effect of the cells, their secretome and their exosomes are enough to make the regeneration in temporomandibular joint regeneration. And lastly, we use the chitosan and hydroxyapatite scaffolds, and we put the exosomes on them in the rat calvarial defect. It is a biological scaffold, and you can see how the exosomes affect and how it makes it greater bone regeneration when you use it with some scaffolds. And I can give some future directions in the oral and maxillofacial region. We have regenerative therapies we broadly categorized as material-based therapies at the first and second generation. And in the third generation is stem cell-based therapies. We are here in the clinic now, but the cell sheets, 3D constructs, and genetically modified cells and immunotherapy probably will be the future directions in the cell therapy. And the bioprinting is very important. You can use them with some biodegradable scaffolds and with stem cells together, and you can produce a viable bone or viable tissues. And thank you for the attention, for your kind attention, and thank you again the organizing committee and Dr. Ulaş Acaros for inviting me. Thank you.
Dear Professor Dr. Zeynep Burçin-Gönen, thank you very much for your excellent presentation. It was very nice. You have put also some videos on site. It was very detailed and informative presentation. Thank you very much again. Dear respected participants, do you have any question to Professor Gönen? You can write your questions also in the chat box, or you can press on the reaction buttons and we can open your voice. And if you are allowed, we can write your email address. Maybe some interested people can contact you by email address. Is it possible, Professor Gönen?
Yeah, I think.
Yeah, it's here.
It's here. It's my Instagram and Twitter, and it's my LinkedIn.
I think now there is no question, but maybe they can write to you directly to your email address.
Okay.
Thank you very much for your nice presentation again and for your contribution.
You are welcome. Thank you.
Dear respected participants, we are continuing now the next presentation, which is going to be held by Professor Dr. Meral Kekeçoğlu. She is from Düzce University, and she is head of the Beekeeping Research Development Application Center. Dear Kekeçoğlu, we are happy to listen you.
Yes, I am here, you, Dr. Ulaş.
Before you have start your presentation,
Okay.
if you are using two monitors at the same time, can you disconnect one of them, because most probably the main problem for opening the presentations caused by that.
Okay. I'm sorry. Everything is okay, Dr. Ulaş? Can you see my presentation?
Yes, we can see.
Okay.
But it is not full screen. It is not full screen now.
Now I did a full screen.
I think the same problem with the previous presentations we has got. You have to continue manually from the left part of your presentation. You have to double click.
Oh, I did. Now in my computer, my presentation is a full screen.
But we cannot see it as a full screen.
I don't know.
Maybe you can you retry it?
Okay. I will try again.
Yeah, please.
Okay. Now it's okay.
Actually not. We are seeing. We can do it so. Can you send it by WhatsApp to me? I can change the slides for you.
Okay, I can try to do. It can be difficult for me, maybe.
Can you change it by you? Can you try to go to the second slide?
Second slide, yes.
Second or third slide.
First, second, yes.
No,
and third, now.
It doesn't change from here. It is a problem.
I get angry, but in my computer, everything is okay. Full screen.
Hocam şimdi siz ekran paylaşımı yaparken aşağıdan share screen yapıyorsunuz ya.
Yes.
Orada iki tane farklı şey var. Bir tanesi slide olacak, bir tanesi PowerPoint. Siz slide'ı seçmeniz gerekiyormuş.
Okay. Yalnızca şey var, paylaş kısmı. O şekilde bir ayrıntı çıkmıyor Ulaş hocam. Yes. Dr. Emine tried to help me.
Hello, Dr. Emine. Can you open in the back side the PowerPoint presentation? Afterwards, come. No, perfect. Just try to go to the next one. We should, yeah, everything's fine.
Okay.
Perfect.
Yes. I am Meral Kekeçoğlu from Düzce University, Department of Biology. Also, I am manager of the Düzce University Beekeeping Research Development and Application Center. First off, I would like to thank you for the good organizing, good congress. Also, I would like to thank you, Dr. Ulaş Acar, for the kind invitation me. Thank you very much for everything. My topic on honeybee venom, correct production techniques in honeybee venom and distinguishing properties of Anatolian honeybee venom. Honeybee venom, known as a apoptosis, is a protein and peptide-rich external secretion produced by honeybee. Because everybody knows that many bees in environmental and every bees secreted the bee venom. But I will talk about the honeybee venom. Bee venom used in many areas, especially cosmetic area, for the mask, creams. You are seeing on the picture creams and tablets and masks, and also lip balms, pain relief, heat patch, and etc. Many products can be used. Bee venom can be used to the bee venom can be used for the many products. Also, bee venom used for the medical area, for the healthy, due to its rich chemical content. apoptoxin has a widespread use in cosmetics and healthy, especially in apitherapy. Everybody knows that apoptoxin can be used for rheumatoid arthritis, ALS, MS, and I'm sorry, Alzheimer, etc. For many health problem, apoptoxin can be used for the many problems. Why correct production factors are important? It's important because B-venom used in medical area for healthy problem. So correct B-venom can be used for healthy problem. Also, it is important the quality of the B-venom because sustainable of production technique is important. Also, hygiene, B-venom used for healthy. So hygiene is important factor. Production amount and colony healthy techniques is important. Techniques is prime factors. Now you are seeing on the picture many B-venom, good quality B-venom, adulterated B-venom, low quality B-venom, and gel B-venom. If you don't use the true production techniques, you can take a poor quality B-venom, poor quality. You are seeing if you don't use the true device, B-venom collection device, you can take gel B-venom. There are no the gel B-venom, not true gel B-venom. So if you want or beekeeper want to do take a good quality B-venom, they can be careful for the production techniques. Also, you are seeing in the picture, number one, a good quality B-venom. Number two, poor quality B-venom. Normally, when you look at the picture, two is a good norm, but it is not good. Color is not better characteristic for determine the quality of the B-venom. Under the light, good quality B-venom is shining and crystallized B-venom. Number one is a good quality B-venom. Also seeing the chromatogram of the this sample on the picture. Sample one's chromatogram and sample two's chromatogram. The factor, many factors affect on the B-venom quantity and quality. The factors, the effect of the device used in the honeybee venom production, you are seeing many different device. This also in front of device and top of the device and inside of the device. You can take the samples, honeybee venom samples with the many different devices, but which one is the better? You can take in front of the hives, or you can put the device top of the hives, or you can put the device inside of the hives. But the device characteristic is an important factor on the B-venom quality. Your device should be different frequency, but the better frequency is the 50 and 100 hertz, or and also stainless copper wire with a voltage range and 12 or 30 voltage is a better. The distances between device is important factor also. The quantity and quality is affected from the device. You are seeing the picture, a gel B venom collected a device, but not true or better device. The device is cannot be used for the collection of the B venom. The other factor effect on the B venom, harvesting site. Before, as mentioned, and first picture, I saw the many device, and also site is important. You can collect the B venom from inside or from top of the hives or in front of the hives. But maybe it's not maybe studies show that the better method inside collection, because in front of the hives there are contamination risks with the pollen. For example, with the honey, there are contamination risks. Due to this contamination, the contamination is the better one inside collection. You are seeing a video in our center collecting B venom in front of the hives. The other video you are watching inside collection. Yes, you can do or you can take with the both of them, but the better one inside collection. Application period is also important factor on the quality and quantity of B venom. Application period must be, our advice is that 15 or 20 minutes. After you put your device, collection device inside of the hives, you continue or you keep your device inside of the hives about 15 or 20 minutes. Other notable factors, production time. Production time, you can take or you can collect the B venom in night or in the morning or in the afternoon. But before our previous literature showed that the better B venom quality and quantity, regarding the quality and quantity, better venom in the afternoon, about 4 o'clock, 4 o'clock or 3 or 4 o'clock in the afternoon. Feeding effect, many researchers claim that feeding is an important factor on the quantity and quality of the B venom. So maybe pollen additive feeding is important and pointed factor on the quantity of B venom. The effect of the other effect of also season. Season is important factor on the B venom quality and quantity. Season May, June, July, August. Between this period, you can take a better collection, better B venom collection. You can do. The effect of honeybee rise and venom quality. Some researchers claim that there are differences between the B venom quantity and quality collected from different rice. The effect of the age of the venom is also other important factor, and storage condition also a significant factor on the quantity and quality of the bee venom. Also, maybe it should be kept in lyophilization form, or it should be kept in maybe must be also in the freeze. Also method validation, other factor on the bee venom. In our center, we did many studies on the factors effect on the bee venom quality. There is our center, and the techniques is important. During the collection of the bee venom, we be careful to do true techniques, useful techniques. Disinfection is significant factors on the bee venom quality. Before the collection, mask, glove, bone, and drying paper, and also alcohol must be prepared before the collection working. You are seeing the picture our center. We use the electroshock-based harvesting device. Also, all samples obtained by active operation of the device 15 or 20 minutes. We repeated harvesting every 15 days. This is important factors. You shouldn't take bee venoms in every day from the same colonies. After you collected bee venom hives, you cannot take again from the same colonies. Collected samples transferred to the laboratory on the cold chain. The collected samples kept in dark glass bottle, minus 20 centigrade degree. Yes, during the collection of the bee venom in our center, cleaning device and equipment moving to the hives as sterile, and also placement of the device in the hive. You are seeing picture 2, and you are seeing picture 3, proper placement of the device in hive. Glass trays will be removed from the hive after 15 and 20 minutes of the application. It is important the time period because of the colony health. Is significant for us. And scrapping equipment must be prepared before the collection, before harvesting. Bone, safety glass, mask, cotton, scraper, and also undercolor glass, glass funnel, and glove. And you are seeing scraper and also our bee venom, and we are taking a photo only in this tube, and then moving to the this amber color glass, and we keep in refrigerator all bee venom. And in conclusion, you are seeing pictures, the step by step harvesting working. Picture 1, we put the device inside of the hives, then it was worked the device forward with the forward device, and then we take off the glass plate from the hives with the bee venoms, then we scrap it, and then we move the glass to amber color glass, and we keep in deep freeze. Yes, in our center, beekeeping center, we did many studies about the factors effect on the bee venom quantity and quality. It's important factor. Before I mentioned many factors, bee rise factors and season factors, and period and time, and storage condition, and many factors. It's interesting that we are surprised because we collected many samples in night time and daytime, and from the inside and in front of the hives, but we are surprised that there are contrasts between the literature. There are no significant differences. You are seeing on the table, there are no significant differences inside hive and front hive, in front entrance of the hive, entrance of the hive and inside hive, inside of the hives collection bee venom. There is no significant differences regarding the content of the bee venom, compound of the bee venom, apamine, melittin, phospholipase, and pH and the moisture. So we did another studies about season. We collected bee venom in May, in June, in July, August. And also there is no significant differences between the bee venom, collected bee venom samples. There is a contradiction between the literature, controversy between the literature. But there are significant differences between the location. We collected many samples from different locations of the Turkey, and we showed that there are significant differences between the samples regarding the location. Yes, also our center. We did another studies on the Anatolian bee venom and commercial bee venom. We compare Anatolian bee venom and commercial bee venom, from different countries, and we show that some characteristic is better than the commercial venom. Anatolian honeybee venom's characteristic better than commercial bee venom. Yes, you are seeing the table on fructose profile, fructose, glucose, sucrose, and many sugar profile, and that investigated that glucose, fructose, sucrose, and totally sugar profile is important for the adulteration. If the bee venom have adulterated, we can investigate looking at the sugar profile. And we compare ordered samples from important internet or via internet. We order many samples and we compare again with the Anatolian bee samples. We investigated Anatolian bee samples at better and good quality, and also we showed some specific characteristic of the Anatolian bee venom. What is the specific characteristic? You are seeing on the table many characteristics for apamine and melittin, phospholipase. You are seeing the mean value, and you are seeing the mean value, also higher value. Since from past time to time, it was surprising that highest phospholipids value we show that in Anatolian B venom, 31, the highest value from past to time. And we compare other studies results, Anatolian B venom and the results, previous results, and we show that also higher phospholipids value in Anatolia. Maybe it might be that is characteristic to Anatolian B venom. Anatolian B venom phospholipids value in Anatolian B venom higher than other B venom samples, and also a little bit lower apamine value in Anatolian B venom than others samples from other country. So I'm sorry. Okay, I'm sorry. Yes, honeybee venom standards. About 17 years ago, Turkish Standard Institute established a detailed standard regarding the content. According to Turkish Standard Institute, the content of the dry honeybee venom samples in mellitin 40-50, between the 40 and 50, and apamine value between 1 and 3, phospholipids between 10 and 12. But in our results, we investigated higher phospholipids value and a little bit lower apamine value. So Turkish standards should be revised according to our results. Apart from apamine, mellitin, and phospholipids content, it is recommended to analyze moisture and sugar. Sugar, maybe it's important because of to investigate the adulteration of the B venom. Thank you very much, your kind and your careful. Thank you very much.
Dear Professor Dr. Meral Kekeçoğlu, thank you very much. It is very interesting about the B venom, and I would like to ask the participants if there's any question to Meral Kekeçoğlu. Yes, Stefan. Can you read?
Yeah. Okay. Now. Okay. Very good presentation. Very interesting with many new details. There is one technical thing I would like to check with you. You put inside the collector the glass, and then it's these electrical wires. But do you put on the glass also a special membrane or not?
No.
Why not?
Yes, Stefan, thank you very much. Your good question. No. But we order device you mentioned. We try to do this device also. In my PhD student's thesis, we will try to do.
Okay, yes. Let me show you this photo. Maybe you see my screen now?
Yes, I know.
Yes, okay. So with this kind of membrane, it's you avoid the contamination of venom because the bees, they make caca, they make they vomit honey or nectar from their mouth, but also there is dust and spores in the air. So, but the problem is with this membrane, this polyethylene kind of membrane. Maybe you can speak with the best Turkish companies which are making these plastic membranes, because they must allow the elimination of the water, humidity, and also must be neutral. No chemicals should come into the venom. Yes. So, yeah. But this is the very good method, because this is the method used in It was used in Romania and in many other countries many years. And then I saw this system with not using it, like in Korea, and then you get a lot of contamination.
Yes, thank you very much, Stefan. We order your device. We know that, and my students tried only one times. Very good B-venom collected. She collected very good B-venom with your device, and white, more white.
Yeah. Yes.
Color
Right.
is more white.
White
Yes.
color. Yes.
But advantage of the inside device is that many device you can put inside.
Yes.
But entrance of the hive only you can put one device.
Yes.
So for the amount of the B-venom is important factor maybe.
Yes. Yes. And also outside it's the sunlight.
Yes.
The radiation which attacks some enzymes. Yes, it's very, very good and very detailed, your work. Congratulations. Yes. Bravo.
Thank you very much for your contribution, Stefan, and thank you very much for a very informative presentation, Professor Dr. Meral Kekeçoğlu. Dear respected participants, we are continuing now with the next presentation. Before that, I would like to ask Professor Meral Kekeçoğlu if any questions is going to be directed to you. They can send you email. If it is possible, can you share your email address in the chat box?
Yes.
Okay, thank you very much. Thank you very much. And the next presentation is going to be presented by Professor Dr. Yanka Karamalakova from Bulgaria, from Trakya University. Dear Professor, welcome and thank you very much for accepting our offer.
Hello?
Now your voice is open. No, now your voice is open.
Okay. Hello, I'm Professor Karamalakova from Bulgaria. I'm from Trakya University. And for myself, I couldn't present nothing about the honey and all this stuff. I present something totally different. I will show you. My presentation will be about one natural compound, lambda minor, extract and the ameliorated properties of the intracellular reactive proteins and Ross levels impressive bleomycin-induced pulmonary fibrosis. I hope that every one of you can see.
No, actually not. First you have to open your presentation. Afterwards, you should come to Zoom, and then
yes,
at the bottom there is a green button.
It's shared now.
It is shared now, but it is.
It's too high.
It is too high. Yes. Maybe you can make it full screen.
Just a minute.
Mm-hmm.
Now it's okay?
Yes, okay, but you have to go at the bottom, the special button showing the presentation.
Where?
At the bottom.
Your screen sharing?
You can put just F5, F5.
F5?
Yeah, F5.
It's okay now? Yeah, I think so. Yeah.
Actually not. Then you can go upside. You can go upside.
Yeah.
And upside, you can see the green button. It is now upside of your page.
Click on that. Or resume share.
Reshare, and just select there the presentation. There are two different presentations there. One of them is the PowerPoint, the other one slideshow. Just select the slideshow.
No, I couldn't see this part of
you on your ecran. You see now the full screen, or
Just a minute. I'm looking. I have a mute, stop video, all these things. New share. And
new share, yeah.
On the new share.
Yeah.
And
there you should select the
this one. Now it's okay.
Now it's okay. Yeah.
I think it's okay.
Perfect.
Okay, ladies and gentlemen, I'll present our work for the last 20 years. We are chemical doctors, and with my colleagues here in the EPR Center, we work about different disease. Most of the disease are progressive disease. We induce this disease by different cancer treatment or by different antibiotics or all these things. And today I talk about induced pulmonary fibrosis. First, I explain shortly why we decided to work with the lamina minor, because the lamina has a really big quantity of the proteins in the structure, vegetable fibers in the structure, fats, polysaccharides, flavonoids, amino acids, aliphatic acids, phenolic acids, and other compounds, especially the vitamins. And Bulgarian lamina, we identified it before to start to work, have a 32 biologically active substances. From this, the photosterols were in a really high quantity. Saturated hydrocarbons were the second bioactive compounds. Aldehydes and ketones were third, and fatty acids and their derivatives were third for importance compound from the lamina. From the literature, we know that the lamina is just a watery flowing compound and has strong antioxidant activity, catalyze activity, superoxide dismutase-like activity, total antioxidant capacity, DPPH absorption, more than 28%. And we decided to use around 180 milligram per kg or 280 milligram per kg to reduce the induced lung fibrosis. And most of the analyses were made by the EPR analysis and other spectrophotometrical and other immunohistochemical analysis. What was the question? Is the lamina, does it affect pulmonary fibrosis in the mice models? The mice models were made by us here for the 2 months because we wanted to know, does the lamina have a protective or have a therapeutic effect? We decided that the experiment will be more than 33 days, but I will present just the first part, what happens after the stop of the 33rd day. For shortly, what is the pulmonary fibrosis? In the down part of the both livers, doesn't matter right one or left one, we have a fibrotic cells that going from the down to the up part. And finally, the animals, and especially the peoples, have really different problems. For that, to ignore all problems and to protect the problems of the idiopathic pulmonary fibrosis, we used the lamina. How we induced the pulmonary fibrosis? By the bleomycin. The bleomycin is a cytostatic antibiotic. It's very bad species, used mainly as a chemotherapeutic due to lack of the after the myelosuppression and immunosuppression. Work about the testicular cancer, Hodgkin lymphoma, but at the same time, in a different dose depends manner, make a really huge problems with the lungs. And these problems with the lungs, if the patient couldn't die after 2-3 years from this kind of cancers, definitely will have problems, and the problems will be with the lethal character from the bleomycin induction. Cytostatic effect, in vivo, in vitro, all the time reduce ferric anions from the second and from the third part, means that all the time it's in an oxygen chain reduction. All the time bleomycin is conjugated with the DNA, and after exposure of the DNA cleavage reaction oxygen and nitrogen species, finally the DNA chain leads to the cell death. Also, the bleomycin induced really high concentration of reactive oxygen and nitrogen species, and the most important, makes different immunoinstability in the organism, destroying the lung cells, destroying the lung architecture. And after destroying the lung architecture, the intracellular tumor necrosis factor, interleukins 1, 6, 18, 21, 70, 17A, plus one different TGF, make a really bad for the organs, for the organisms, and of course for the patient. Because of this, because in the medicine we use the bleomycin, now we decided to make the same with the animals and induce the bleomycin-induced progressive pulmonary fibrosis for the 33 days. On the 16th day, we take one of the groups of the animals and give to our colleagues in the anatomy department. They make an experiment there with the immunohistochemistry and with the mast cells and look at what happens. And on the 29th day, we stopped with the bleomycin induction, and definitely with lamina minor induction. And on the 33rd day, we sacrifice the mice. And when you take from the mice the liver, you could see what happens here. First, the liver in the control, the group that didn't affect it with nothing, just the fresh water and normal tap. Here, the bleomycin induction, you could feel the lesion and the mast cells lesion here, very important and very effective from this antibiotic. And after combination by the lamina, we'll look at almost the same character of the liver cells and the liver as and the lung, as the character and architecture here as in the control. We look at the doses that we induced about for the pulmonary toxicity and induction of the idiopathic fibrosis. And we make it really different experiments, but the most important one is that we induced every single day, 2 hours before the bleomycin, the lamina extract. You look at here, 200 milligrams or from the 6 we make injection because more of the here our colleague going to this, and we have first group controls, second one bleomycin administration, third group lamina treatment, fourth group lamina treatment and protection under the bleomycin against bleomycin incorporations. After 33 days, we sacrifice the mice with the very easy one method by the Nembutal as a drug. Take the lung samples and take the blood samples and analyze for some different kinds of parameters, catalyze activity, SOT and glutathione peroxidase activity, and after that, MDA activity, lipid peroxidation, and look at what happens with the protein carbonyl content AGE factors, ROS levels, NO levels, or we'll look at what happens in the protein or carbonyl content, what happens with the proteins after 33-day administration of the bleomycin. Is it possible to lamina to reduce all reactive oxygen and nitrogen species? And after that, to make the proteins to react again easily without completers. For this purpose, as a result, we were really excited when we see that we have a superglutathione, long superoxide dismutase and catalyze activity. We could look at the lamina minor activated a bit the antioxidant system in the body and look at what happens with the same enzymes activity after bleomycin inductions. Almost nothing from the antioxidants in the system. And a combination, a really good positive effect about the body. Even in this kind of concentrations, 200 milligrams per concentration per kg were enough to reduce the antioxidant system, to reduce the enzymes in the body, and to twofold increase in lipid peroxidation, again, in the lung, and to reduce carbonyl proteins and look at in what kind of statistical differences. The second result, by hydroxyproline analysis. Hydroxyproline, it's the special compound who all the time going to reduce the structure of the lungs and going in the dark part of the lungs in a huge quantity. Because of this, we all the time make an analysis about the hydroxyproline concentration in the different organs, doesn't matter, lung, liver, or some other kind of organs. And by the concentration of hydroxyproline, we could know if we really have bleomycin induction of the idiopathic fibrosis or not. By our results, here you look at the controls, we have 2 times higher induction of the idiopathic fibrosis. And we look at the lamina minor concentration, it's almost as the controls. We didn't take nothing, especially the normal food and normal water. And the combination, lamina have really protective effect and therapeutic effect for the 33 days. The lamina reduced the bleomycin induction of the hydroxyproline almost 2 times, and 2.7 times reduced the hydroxyproline concentration, means that the lamina reduced the lung architecture almost 3 times. In another analysis for protein carbonyl content, for glycation and products in 3-methylpropyl protein oxidation, I want to explain something about the methylpropyl. The methylpropyl is a spin trap for trapping in the blood samples or in the urine samples or in the organ samples, the organ homogenized after homogenization, the small quantity of the protein oxidation means the small quantity of the albumin reoxidation. This spin trap by the APR spectroscopy could be defined what is the concentration. First, we make the protein concentration. We look at again a really huge protein concentration in the lungs. After that, we make in the blood also. After bleomycin induction, for the 33 days, the animals almost didn't have the lungs. After that, you see that the lamina, the waterland, didn't make the proteins, didn't concentrate the proteins, and didn't analyze the proteins, just reduced the protein carbonyl content. And this is really visible in the combination on the bloomicin group. About the aggregation products. The aggregation products, it's a really good characteristic about the protein carbonylation content. And again, we'll look at the same results. So it's totally normal when you have a really huge concentration of the protein to have really huge glycation end products. And here we'll look at in the IgG that the lamina reduced a bit in IgG products also. In the last one, about the concentration of the albumin in the lung samples, in the blood samples, what was important? Here, the EPR spectroscopy crossed the electrons and all the time catch the oxygen levels. If you see here, we have a really huge concentration of the reactive oxygen and nitrogen. Means that the albumin concentration, it's almost 1%, means almost 100%. If you look at in the tube, in the organs, and here the lamina reduced almost 3 times the concentration of the short albumin, reduced the CH, an important group, and reduced the different cooperation of the structure of the proteins, which were really important for us. Because of the situation of the ROS products, we directly examined the reactive oxygen species by one spin trap by the PBN. And what happened? The same results. Again, the bleomycin, it's the bad guy. Huge concentration of the lipids, of course. Huge concentration of the superoxide anions, who after time crossing to the OH anions, to the hydrogen peroxide anions, and after that to the lipid peroxides who stayed in the organs, and lamina without this kind of disease. And you could look at here in the combination, the lamina reduced in a really huge manner, in a dose-dependent manner, in a time-dependent manner, reduced the bad reactive oxygen species concentration. And here we could say that lamina has not only the protective but the therapeutic effect, because for the medicine, protective effect, it's a good thing, but therapeutic effect, when you start with some kind of disease exposition, we could look at which one of the oxidants, doesn't matter vitamin A or C, could protect the disease, but which one of the natural compounds plus vitamins could have a therapeutic effect. And for the other group, with the patient will start with this clinical treatment and everything will be okay for the second, for the third group, because you can reduce the dose, or just you could explain the more has dosed with this natural compound. Something else, let's just look at. We have a habit, after all these structural compositions about the homogeneity of different organs, finally to make immunological analysis. And we always make one and the same immunological analysis about the interleukin-1 beta 4/6 tumor necrosis factor. And we make at the end, on the 16th day, means the day when you look at if it's possible to have a fibrosis in our mice models, and on the last day before the sacrificing. What we see, our question, does it lamina minor acting as a protector and as a therapeutic agent, have an answer? And the answer was yes. Yes, the lamina acting as a protector, even on the 16th day, even not only in the lung homogenates, but in the blood also. But the most important was that on the 33rd days, the lamina minor acting as a typical therapeutic effect, therapeutic agent. And this is seen about in all of the interleukin expression, means in the cytokine expressions, and this is seen about not only in the liver homogenates, but in the blood also. Now I'll repeat some of our findings. We found that bleomycin stimulate the concentration and time-dependent interleukins, secretion, and positively correlated with the histological findings. What we found that the mast cell number were more than 1 million, and a composition structure more than 1 million in the liver with the bleomycin. We found that have a really high concentration of the collagen bleomycin deposition, means the hydroxyproline deposition, and the protein synthesis in the lung tissue in all studied group except the controls were really, really high. Although the lamina minor effect on the bleomycin induced correlated early and late inflammation and fibrotic processes are due to the decreased mast cell number in lungs, which lead to the decreased cytokine concentration in time-dependent manner between the 16th and 33rd day. We have a really good deposition in the lamina, in the lungs, in the blood, and especially at the 33rd day. Since administered bleomycin-induced inflammation is more efficient in the lungs, the mast cells clearly may be prominent. Actually, mast cells are able to produce and release all of these interleukins. The mast cell release histamine-induced pro-inflammatory cytokine production, including interleukin 6 and 8, and of course the tumor necrosite factor L-alpha. Also, TNF stored in the mast cells granulates activated macrophages, endothelium, and cytokines by the endothelium cell binding and leading to increased adhesion molecule expression. The activated leukocytes are able to bind with the adhesion molecules and then evolve the inflammation site. On the other hand, we have one of the colleagues who work with us, not in Bulgaria, of course, the DCN who emphasized that interleukins 6 and 8 increase the mast cell proliferation, the same that we look at here, and the more active factor formation by the activating directly through the interleukin 6R and GP130 receptors expressed on the mast cell cellular certification. The mast cell cellular maturation leads to increased granularity, cell size, and hemolase content, and the enhanced reactivity was evident from the increased cytostatic mediated granulation and cytokine production. Also, interleukin 6 blockage may ameliorate the mast cell related symptom and pathology in the patient with mast cell related disease associated with the increased EL6 levels. Taken together, this result indicate that our lemon extract can stimulate fibroblasts to reduce collagen deposition and albumin synthesis in the mouse alveolar epithelial cells and contribute to tissue restoration in a time-dependent manner. However, when you consider that the fibroblasts were apparently separately, were detected from the 16th and to 33rd day, and the oral anti-inflammatic and anti-fibrotic levels of the lamina may be the result by the systematic inflammatic infection, blomycin-induced inflammatic on the mast cells and other alveolar epithelial cells. And what was our hypothesis? We hypothesized that lamina antifibritically activated and promote the acute or chronic inflammation blocking, resulted in a shift from the immune response in the fibrotic lungs down Th1, Th2 responses, very important responses for the body, for the cells. When you listen to cells language, this is more important responses for us, and effectively induce the activation of the collagen and albumin degradation. And for that, lamina show considerable efficacy against interleukin-1 beta, 6, TGF, TNF-alpha. Oxidative stress may be associated with the early changes between 60 and 33 days, maybe even between 16 days in vascular parenchyma in the idiopathic pulmonary fibrosis. What to say? This was really unexpected for us. Why? Because we didn't expect that the extremely small compound with extremely small size, water length, something really unimportant, will have that extremely important effect. Thank you. If you have questions, I'll be glad to answering, but if it's not in your area, I'll understand.
Thank you very much, dear Professor Yanka, for your nice presentation. Is there any question to Professor Dr. Yanka about her presentation? Actually, my brother has also worked with lamina minor during his master thesis. It was nice to hear about this subject again.
By the way, I have an answer from the antioxidants, and this will be as a final, one of the final my works, and will be as an article with the impact factor 6.9, and will be on the internet after 1-2 weeks. A really good result from our work. And I hope that everyone will have like this success.
Thank you very much, Professor Yanka, for your presentation. Maybe the researchers would like to make some contact with you. Therefore, if it is possible, can you write your email address on the chat box?
Okay.
Thank you very much. And dear respected participants, we are going now with the next presentation. I have called Professor Dr. Raul Pascao, but he has got sudden health problems. That's why he is apologizing us. And we will continue now with Professor Dr. Galina Nikolova from Trakya University Medical Faculty. And we are happy to listen Professor Dr. Galina.
She is here.
Okay.
Hello.
Hello, dear professor.
Just a moment, 1 minute.
By the way, dear researchers, the poster presentation section is going to be done on Sunday. I have written it in the chat box. Also, additional email was sent for the poster presentations.
I'm sorry, just a moment. I will open my presentation.
Okay.
Here you are. Thank you very much.
My name is Associate Professor Dr. Galina Nikolova. I work with Yanka in one team. My presentation is on topic gamma irradiation effects on food long-term storage. We studied Portulaca oleracea extract by gamma radiation on 10 and 20 kilograms. In recent years, food irradiation has been used to protect against oxidative toxicity, and this methodology is internationally recognized for effective long-term storage. This is according to the directive from 1999. What we know about Portulaca oleracea or purslane, about scientific classification is from Kingdom Plantae, clade Technophytis, order Caryophyles, family Portulacaceae, and genus Portulaca.
Dear Professor, sorry for interrupting you. Can you share your presentation? We cannot see it now.
I'm sorry. Oh, this is my mistake. But I have information that I share it my screen.
No, we cannot see it now. First, you have to open your PowerPoint presentation. Then afterwards, you have to come to your Zoom screen. And in the Zoom screen, you have in the bottom there is a green button.
Share screen.
Share screen. Then click on it. Then select your presentation.
Can you see it now?
Actually, not.
The screen is green.
And now it is open. Please go bottom of your presentation and click on the icon. The next one. Yes, now, and just try to go further. Yeah, everything's fine. Thank you.
Okay. Yeah. Okay. Thank you. So, about gamma irradiation effects on food long-term storage, about Portulaca oleracea extract. As I explained, the scientific classification of Portulaca oleracea, it is from Kingdom Plantae, clade Tracheophytis, family Portulacaceae, genus Portulaca, and species Portulaca oleracea. It is a green herb containing omega-3 fatty acids, gallotannins, camphorol, quercetin, apigenin, glutathione, and vitamins. And the stem and leaves with high energy values, about 303 kilocalories on 100 grams, and minerals as potassium, sodium, calcium, iron, zinc. Contained proteins, also lipids and fibers. Portulaca oleracea phytochemical is a phytochemical composition and representable for its biological effect. It has high antioxidative, antiseptic, antibacterial, anti-inflammatory, anti-asthmatic, and anti-radiation effects. The leaf from this vegetable I use in salad or cookies. It as spinach, because of its mucilaginous quality, is suitable for soup and stews. It has a slightly sour and salty taste and is eaten throughout much of Europe, Middle East, Asia, and Mexico. The stems, leaves, and flowers, buds are all edible raw and all cooked.
Nutrition raw purslane contains 93% of water, 3% of carbohydrates, 2% of proteins, contains negligible fat. In a stock, in 100 grams, a reference amount purslane supplies 20 calories, and much amount, or according 20% of vitamin E, 81% of vitamin C, and according 19% moderate content of several dietary minerals. Richest vegetable source of alpha-linolenic acid and essential omega-3 fatty acid. To study a lot of studies shows that the Portulaca oleracea have inhibitory effect on lipopolysaccharide on interferon gamma, induced NO production, protection against oxidative stressors as 9-kg radiation. Portulacacea from Bulgarian flora, doesn't it have a protective effect against, according 10 kg and 25 kg radiation? This is our very important question. And to answer to this question, we studied Portulaca oleracea after irradiation. For this, we studied dried samples which were irradiated on 60 cobalt dosimeter biomax at 8,200 for 1.37 minutes directly. Powered and extracted by 30% ethanol solvent to 88% purity. We studied this is extract with EPR electron paramagnetic X-band resonance spectrometer on Bruker. I see promising techniques for foodstuff identification, antioxidant capacity, and regulatory requirements was used. To study the Portulacacea, Portulaca oleracea, we use the spectrophotometer. It's the structure is presented here. We situated the sample in in a cuvette and measured the spectra, rated the spectra on different kilograms. As you can see, on 0 kilograms, the G factor is according 2.0023. This is the spectra of extract on Portulaca oleracea on 10 kilograms. Irradiated, and here you are the spectra on 25 kilograms irradiated. The difference comes in 10 kilograms. It's much more effective, closer to this, which is on 0 kilograms, which is placed as our control. The ability of Portulaca oleracea to scavenge in vitro short-lived radicals with spin traps, we examine it by using spin trap BMPO 5-terto-butoxycarbonyl 5-methyl-1-pyrrole-N-oxide, tempo 4-hydroxy 2.6.6 tetramethyl-piperidine-N-oxyl, and DPPH, diphenyl 1-picryl-hydrazil. Here you are the results from BMPO examination. Again, we have a difference between the control, 0 kilograms of extracted extract from Portulaca oleracea. Again, we have close spectra on 10 kilogray irradiated portulaca oleracea, and in 25 kilogray irradiation of portulaca oleracea, we haven't got this a good presentation of spectra. They are much more like sound, like a sound, and the same result we have with portulaca oleracea and studied with tempo. Again, the 10 kilograys irradiation was close to control, and 25 was with the less result. With DPPH, we have with 6 spectra line in control, and only one main line on 10 kilogray portulaca oleracea irradiated, but with the same intensity. According to the 25 kilogray irradiation, the intensity was less, and we haven't got so good presented main spectrum. What we can say as conclusion, the kinetics of 0 kilograys, 10 kilograys, and 25 kilograys irradiation induced EPR signals was studied for a period of 50 days and 6 months post storage. It should be pointed out that stable radical structure were registered for the mentioned period for 10 kilograys irradiation. In the case of adduct formation, the signal intensity increased for portulaca oleracea after 10 kilograys due to third month. The high dose irradiation, 25 kilograys, statistically significantly decreased the ROS scavenging, adduct scavenging of portulaca oleracea. So we can conclude that maximal dose for foodstuff identification, protection, and antioxidant capacity is 10 kilograys. And thank you very much.
Dear Professor Galina, thank you very much for your nice presentation and for your support and contribution to the Congress. Dear respected participants, is there any question to the Professor Galina Nikolova? You can write your question in the chat box, or you can press the button for the reactions, then I can open your voice. Dear Professor Galina, maybe some researchers would like to make contact for
I
you.
can write my email, university email.
Okay, thank you very much.
And if someone have a question, I will answer immediately.
Thank you very much, dear Professor. Dear respected participants, now we will close the section, and in 3:30 we will continue. During meanwhile, we will open the poster presentations. You can enjoy it, and the other important thing is the poster presentation section is going to be held not today, on Sunday. Thank you very much again for your participations.